I have to give so much credit to Brittany. I'd say Brittany did about 65% of planning this event. This was a pretty big endeavor for the two of us, and it was the outreach. I'm really excited that so many folks are here. So Brittany Schappach received her master's in entomology from the University of Maine, where she received the Fred Griffey Memorial Award for her interdisciplinary research on black-legged ticks. Brittany currently works as an entomologist with the Maine Forest Service and helps educate the public on forest health threats, including the spread and potential impacts of jumping worms in Maine forests. I suppose we still have 2 minutes here, Brittany, so I got the timing really good there. Maybe if you just want to, maybe you could give some fun field anecdotes some fun field stories or something like that.
Some fun field stories. Oh man, I didn't prepare for this. Maybe we go around the room and tell fun facts. I'm just kidding. There's too many people for that. But can you can see my screen okay? And if I click on it, you can see the laser pointer too, hopefully. Moving around, maybe not.
Yes.
Cool. All right. I love being able to do that. So because I can't point when I'm giving presentations in person, I can usually do that, but here we're a bit limited, so I'm glad. Yeah, so thanks everyone for tuning in. Really excited to have a good lineup for you. If there's at any point you have any questions, feel free to throw them in the chat. Each presentation is going to have our own Q&A section at the end, so you don't have to stay to the end. If there's just one or two presentations that seem really interesting, you'll have an opportunity to ask those presenters directly your questions if you have any. So that being said, yeah, thanks for coming. My name is Brittany Schappach. I'm an entomologist with the Maine Forest Service. Today, my presentation is going to be jumping worms in Maine. And when I was creating this presentation, I really tried to pick out the things that I think most people would want to know about. And I'm trying to leave, there's of course so many things to talk about when it comes to jumping worms, but I'm really trying to leave the aspects of plant cells and all the emotions and citizen science and all the other good things we have.
Sorry, Brittany, I muted you.
That's okay. Everybody, there's a little pickup that says muted. So
if everybody could please stay muted, that would be great. It doesn't make it easy for us to find who's actually muted.
Yeah, we'll have to rethink how to make these lobbies when it was this big. Again, we didn't really expect this big of a response. So again, thank you for being here. So without further ado, I'll just go ahead and jump in. It does look like I've rambled on enough that we're at the 1:15 mark. So I'll go ahead and get started. So first things first, before I dive into jumping worms, I want to talk a little bit more about what earthworms are because they are critical to our understanding about how jumping worms as a whole function. So first things first, earthworms are invertebrates, meaning that they don't have any backbones or anything like that. They don't have any bones at all. They're known to be very wiggly and slimy, and they eat decaying roots, things like leaves, bacteria, fungi, even wood sometimes when it's decayed. And this is the case for all jumping worms or worms in general across the world in all kinds of soils. So I put on here some images of worms that might look familiar to you. That being said, I am giving a talk today about the earthworms in Maine. So this is going to be a little bit more geared to our Maine attendees here. And so there are no native earthworms in the state of Maine, and that's actually the case for much of the northern United States, and that's because of this ice sheet that used to be covering the states. And so when this glaciation event happened many, many years ago, 10,000 give or take, it killed any of the earthworms that were present. And when it melted, we had these soils that never had earthworms present in them before. So that's a good thing to keep in mind. And what happened when this ice sheet basically melted is we actually had the native earthworms that were in the southern parts of the United States move into these areas into the Northeast, including Maine and Connecticut and Massachusetts and things like that. So with this northward expansion, you have the invitation for these worms to be in places where they normally weren't. And so we have all of our plants and our trees and our species that are native to these areas that didn't necessarily evolve with jumping worms or worms in general. So the question on everyone's mind, what are jumping worms? So there are 3 different species that people are referring to when we say jumping worms. You might also hear these as crazy worms, snake worms, Alabama jumpers. I've heard a lot of different iterations. And so when people are talking about those, they're talking about the jumping worms. I do see in the chat that maybe there's some people struggling with seeing my screen. Is that the case? Or maybe it's just this one person?
I can see your screen just fine.
You can see? Okay. All right, good. I just want to make sure because I saw it, so I wanted to stop there. All right, yeah. So whenever people are talking about crazy worms, snake worms, Alabama jumpers, they're usually referring to the jumping worms. That being said, they are non-native and invasive in the United States, which essentially means that, again, they didn't grow up with our native species here in the state of Maine or the Northeast. And invasive in this case is typically meaning that they cause some kind of damage. So ecological, human health, or environmental. In this case, we're going to be, again, focusing on Maine here. And so when these worms are in the soil, they're very different than the earthworms that can be found in the state of Maine. So these are typically found in the top 2 inches of soil, but sometimes they can be found as far as about 4 inches or so, but they're going to be on that top layer. And so these are technically epigeic worms, meaning that they don't tunnel all the way down like some of the other earthworm species. You might be aware that the European nightcrawler or the common nightcrawler can go as far down as 6 feet sometimes. That's not the case with jumping worms. They are going to just be on those top layers of soil. So in the state of Maine, 2023 is actually the year that we really consider jumping worms to be a widespread problem. We had a lot of reports come through in that year. Prior to that, we had noted it in the state, but we did not recognize or really understand how widespread that they were until about 2023. So if we're looking at this image here on my screen, this brown color denotes all of the confirmations we were able to, we gathered through public reporting in 2023. There's a lot of brown on this page. So we had a lot of reports. And prior to that, we have all these other years. It was a few towns here and there, but it really wasn't much. Last year definitely slowed down a lot. We were able to confirm another 7 towns in 2024. And I will throw a link in the chat to a bigger photo of this map for 2024 for those folks who are interested, just so you can actually zoom in and see towns and things like that. So this map is on the DACF webpage, so it's a good resource. But we did start to see the presence in the state of Maine really slow down. So to better understand where they are, what you can do about them, it's also important to understand their life cycle. So these jumping worms are only living for 1 year. So some folks will refer to them as annuals. And when that is the case, their whole goal is to reproduce. They grow really, really fast and then reproduce as soon as they can. So when we're thinking about identifying jumping worms, the first thing we want to do is think about where they are in their life cycle. So right now we're about mid-June, and we're still a little bit too early to be able to correctly identify jumping worms as such. And that's because they are in this juvenile stage in May and June. So they're very small and they don't have this reproductive structure. They don't have that collar, or what we refer to as a clitellum. They don't get that until later in the season. So if you have a bunch of worms that are really wiggly and they're really small, we normally can't correctly identify those until a little bit later in that season. So that's important to keep in mind. The other thing is they are parthenogenetic, which essentially is a fancy word to say that they reproduce on their own. They basically clone themselves. And with that being said, it then only would take 1 adult or 1 cocoon to start an entire new population in infested soils. And these cocoons are present in around November through April, and you can think of them just like eggs. They will hatch in about May time. Again, we're in June, we're in this juvenile stage, And then they will eventually grow enough to become the adult form. They'll get that clitellum. I'll talk more about that in a few slides. And they will die typically the end of October in Maine, and really anywhere in the United States. We are seeing this warming weather pattern. So they are living longer, which means that they have more of the ability to successfully reproduce and make those cocoons. So that's important to keep in mind as well. Let's see here. So when we're thinking about jumping worms, the adults are the problem, right? Because they're doing all of this chewing really, really fast on our soils, but it's also the cocoons that are an issue as well. These are very, very small. I have a photo here that has 3 of the species that are commonly found in Maine. They're pretty indistinguishable from each other, and they're also pretty indistinguishable from just a piece of soil. So they're very, very hard to find. And with that being said, they're very, very hard to know that you're accidentally spreading them. They can stick to gardening tools in the bottom of your tread on your tires or your shoes and things like that. So they're quite easy to spread if you're not thinking, I don't have worms on my gardening tools or my boots, but maybe you're not necessarily thinking of the cocoons as well. There's also this idea of seed banking. And when we're thinking about that, essentially what that means is that in an area when there's a lot of cocoons present, not all of them will hatch at the same time or even in the same year. So that means that you could potentially remove all of the adult worms from an area, and you will still have these cocoons that will hatch throughout the year. And you're still going to have these cocoons that are, maybe they don't hatch one year, but they're still there and they're still viable. They'll hatch the year after or the year after. So there's really this opportunity for this snowball effect of all of these worms that really have a difficult time leaving an area once they're there. So some things you can look for, this is not specific to the state of Maine, but these are photos that I took throughout my field experience, just looking for jumping worms and helping folks with them, is that they eat the soil so fast that it dries out the soil and it turns it into this very, very loose, dry texture. And I've heard it referred to a lot of things. So I've got coffee grounds, nerds candy, ground beef consistency, any of these kinds of things. If you're seeing that in your garden or on a forest floor, they should be sort of starting to raise that red flag. There might be something else going on here. The other thing is you might start to see tree roots becoming exposed as well. And that's because this soil is so loose and dry, it's very easily swept away through walking paths or rain events. These are some more photos that I took. This is a forest setting that had a lot of jumping worms. So you can see sort of in the bottom corner here, there's a disruption of that leaf litter. So it's really just loose soil here. And again, it's very crumbly, it's dry. It doesn't look like good soil, and there's almost little to no leaf litter at times. So if you're starting to see these signs of soil that might have this coffee-like consistency, but you're maybe not seeing any worms right away, and maybe it's a lighter infestation, one thing you can do is use mustard as a detection method. Essentially, you mix a 1/3 cup of ground mustard into a gallon of water, you shake it up, and you can remove the leaf litter or pour it right onto the soil itself, and it'll seep into the soil, and the ground mustard's really irritating to the worm's skin. So what'll happen is they will move away from that irritation and they'll just kind of wiggle up to the surface. This isn't going to kill the worms, so this isn't to be used as something to try to reduce populations, but rather if you are unfamiliar, or not unfamiliar, you're just not seeing any worms in the area, this could be a good way to bring them to the surface so that you can actually identify them. It's also very unlikely to harm most plants and animals, so it is safe for those kinds of things, but you are going to know your gardens best, so use your best discretion. In a forest setting, if you were to do this, I actually took this photo. What I would do afterward is actually cleanse the area with a gallon of just clean water, just to really further dilute that mustard, just to be on the safe side. So how do you identify a jumping worm? So maybe they rise to the top and now you're hand plucking them out. And what are you looking for? So this is what I was mentioning before. This is a clitellum, or their collar. And this is easily seen again in the later season. So this is difficult at this time to see because we're still early in their life stage. They're juvenile worms. They're quite small. They don't have this collar developed yet. But what you are looking for is the smoothness and the flatness of this collar. It can range from milky white to a gray color, but most importantly, it fully encircles the worm. So it goes all the way around, okay? And it's found on segments 14 to 16. And when we're talking about segments, I am counting from the mouth of the worm all the way until the very start of that clitellum. And if it is a jumping worm, then oftentimes they will start anywhere from segment 14 to about 16. And again, these younger worms are pretty difficult to identify. Now, a common night crawler can also be accidentally misidentified as a jumping worm, but they have a very specific clitellum. They have what's referred to as a saddle-shaped clitellum. So it does not fully encircle the worm, does not go all the way around. Instead, it just sits on the top of the worm. And so if you're seeing a worm that might have that collar that looks like it's closer to the head, but you flip it over and you're seeing this clitellum that doesn't go all the way around, that's more likely to be a night crawler or a European earthworm. So not a jumping worm. So these are more photos that I've taken of jumping worms. Again, we're looking for this milky white color. Sometimes it's gray, but it's usually flat. Later in the season, in October, late October, sometimes the clitellum can be a little swollen, and that's because it is a reproductive structure for them. And so if they have just recently or are going to lay those cocoons, those eggs, sometimes it can look a little bit puffy. But again, I'm checking all the way around the worm. That clitellum's meeting at both ends. And so I was able to identify these if maybe counting the segments is a little bit too hard because they are quite wiggly sometimes. The other thing you can do is actually check the hairs. This is very specific to all three of the different species of jumping worms. Each segment, it has bristle-like hairs. And so, for instance, this is a photo of a nightcrawler. And this is what's referred to as widely paired hairs. So you can see here, there's just two. It does not go all the way around here. They're just little segments of these little small hairs. I don't have a photo of an adult jumping worm with this hair structure, so I don't have anything to show you with the bristles, which is common for the jumping worm. But if you were to imagine these little tiny hairs that go all the way around the worm, if it didn't have a clitellum, but you're seeing this kind of structure with a hand lens on the worm, that could be another way to identify it. But again, if you have not dealt with jumping worms, they are very, very wiggly. So this could be a more difficult way to identify them. And like I was just saying, these worms thrash about. They got the term snake worms or jumpers for a reason, right? So they don't actually jump. They don't have hind legs. They can't, really like flip themselves out of buckets or anything like that. But they have this nose to tail movement that they do that really helps them pop out of your hand or out of a bucket or off of a surface. So it looks like they can be jumping. Occasionally they can shed their tail if they're really threatened, but European night crawlers can also do this, so I don't typically use this as a way to identify. So as far as impacts go, really is not great for our soil here. Their castings, or what we refer to as frass or poop, is very nutrient dense, but because it's on those upper layers of the soil, it's not deep enough for any of our native plants or trees to actually use that nutrients. And it happens so fast that it's not really beneficial nutrients for our native plants. That being said, too, there's a lot of fungus root relationships between our native plants and our soils, and because of the fast turnaround of these jumping worms, that also often gets disrupted. There's also things like erosion. Again, I was saying it's very loose and dry castings, easy to wash away into runoff. You can start to get some tree root exposure here, which, as you can imagine, is not ideal for our native plants and trees. It's a very stressful event and could result in mortality or stress. And this is a nice summary. There's so much to this concept that I want to leave to the other speakers too. So I'm just briefly going to touch on some points. But there's a decrease potentially in plant diversity in our native plants and insects. Again, this healthy tree root system that could be lost as a result of these jumping worms quickly moving around the soil. There's also lower leaf litter as well, because again, they're chomping at everything, they're breaking everything down, and they're not really releasing it back into the environment in a way that things can reasonably use. And with this being said, if we are having these root structures disrupted, our native plants and our trees are starting to struggle, they might succumb to mortality or stress or other insects that are going to really take advantage of the fact that they're struggling. And they might die. We might see tree and plant mortality. And with that, we have maybe a disrupted forest ecosystem as well. So other plants, animals and insects and things like that, that could be disrupted from this. And if we are starting to see an increase in forest damage, so we might see our forest trees and our forest plants sort of start to decline, it might create an opportunity for invasive species to really come in and take over and outcompete our native plants and resources. So these are 3 examples, at least in the state of Maine, that are invasive. They can cause some kind of damage to our forests. We don't necessarily want to encourage their growth here, and that they're not great. They're going to outcompete our native plants. I do have a red star here on Japanese barberry, and that's because there is a positive correlation between Japanese barberry and the presence of black-legged ticks. Especially in the Northeast, we don't want to encourage black-legged tick habitat, but unfortunately, the Japanese barberry really creates the right microclimate for them to thrive. And so it can be more than just, oh, the soil is changed. Well, there's a lot of other impacts because when we're thinking about soil and the impacts on trees and plants and animals, we have to think of it as a whole. It can't just be soil alone, although it's a great part of it. As far as the state goes, jumping worms are illegal to import, but they're not a regulated invasive species. So the Department of Agriculture, Conservation, and Forestry, which is us who helped put on this show today and tomorrow, we can't do anything beyond educating and outreach, unfortunately. But that being said, we are working with a whole bunch of different agencies. We have a pretty good working group that meets every single year, a couple times a year, and we're really just trying to put our brains together to try to figure out what people are working on and how we can help and what kind of data sets would be helpful. And one of those is looking at some sites in the state of Maine, at least, that could be good for long-term monitoring. So that being said, I am helping to run that program. If you have jumping worms present in a forest setting, and you might be interested in cooperating with myself for potentially a long-term monitoring site, please send me an email. I'd love to chat and see what we can do for you. The other question that's on everyone's mind is pesticide use. There's currently no products registered for jumping worms in the United States in general, so there's nothing on the market that you can legally purchase for it. And it's important to note that any kind of pesticides for pests that are being used when the pest is not listed on the label is not great and can have unintended consequences in addition to being illegal. One thing that we can recommend for folks if you do have jumping worms in a garden or a large bed setting is this idea called solarization. And so essentially we know that the adult worms die at about 88 degrees Fahrenheit and the worms and cocoons die at about 104. So there's this concept of basically making a solarization package where you put out a thin layer of the soil or mulch or anything that you have that's infested with cocoons and the adults, and you package it up really nice like a little present, and you bake it in the sun for at least 3 days. And the idea behind that is to contain the worms in an area where they will get those temperatures that are reaching their upper mortality limit. So we'll start to see those cocoons die, become not viable, and the adults die. And you can then reuse that soil or reintroduce it into areas, and it's essentially cleaned. Now, that being said, the levels of reinfestation, if you were to solarize, are quite high, especially if you are putting them back into an area that already has jumping worms, cocoons, or the actual adults themselves present, because they will just wiggle back into it. But this can help support certain kinds of native plants or crops or things like that, just to try to reduce those populations. The other thing is hand removal. So you can actually take them out. This would be good for smaller infestations. Like if you have a raised garden bed, you can actually pluck out the worms and drop them into a bucket of soapy water for a bit. And that will kill the worms. And then you can just solarize the soil that's left in the bucket if you want to strain it off, or just bag the worms and then throw them out. But again, reinfestation is quite common in this kind of scenario. So it potentially can reduce the amount of cocoons that get introduced into that area, because you're killing the adults that would be making them. But this isn't going to reduce the cocoons that are already there. The other thing is tilling. So tilling soil in May, if you do have those flat garden beds or an area with the soil where tilling is appropriate, that can actually really help break up those juveniles, and you can start to see reduction in population that way as well. The thing that we really focus on at this point, since research is ongoing, is prevention. So things like arriving clean and leaving clean. Like I mentioned before, their cocoons are so small, and it's so easy to go and put on your nice hiking boots and accidentally bring along those jumping worm cocoons from different trails to your home, to your backyard. So it's really just important to make sure that you're not ignoring that might be a possibility. So these are boot brushes that I highly recommend. I have them in my work truck. We all have them in our work trucks at the state of Maine. And this is, again, it's just trying to brush off any of those cocoons or pieces of soil from the bottom of your shoes to try to not bring them into new places. But it's the same thing for things like tire treads and whatnot.
5 minutes.
Thanks, Hillary. The other thing is, so we're not trying to purchase jumping worms for purposes of gardening or fishing bait or any kind of composting. Unfortunately, we have seen instances like this photo here of people using them as fishing bait, and then they didn't finish the rest of the cup and they just dumped them into the woods. And then again, it only takes one cocoon, it only takes one worm to start an entirely new population. So this whole area is now infested with jumping worms, unfortunately. And the other thing is you really have to be quite smart in how you are discarding yard waste and anything like that. I would treat anything, any kind of leaf mulch, soil, that kind of thing. If you know you have jumping worms, treat it as if it's an active, ongoing infestation. So please don't introduce it into our forests. But do teach others about jumping worms. So we do have a really great turnout today, but there's a lot of other people that just don't have the capability to log on to things like this, and maybe they're not as connected here. So it is important to talk to your friends and your family and your neighbors. Just spread the word. Let people know what's going on. Let people know what they can do. And also that more and better research is ongoing and coming. An informed buyer is also really important here. There's a lot of questions from our initial survey of, how can I purchase composting soil or mulch or things like that? How do I know if it's safe? The question is, you don't really. The best thing that you can do is ask if it is heat treated. If it's heat treated, they are essentially like super solarizing it. And so it should be able to kill the jumping worm adults and cocoons if they're present, along with other nasty invasive stuff that we don't necessarily want to introduce. So heat treated is a really important question to ask if it is not advertised on the bag or at the nursery that you're buying these things from. And choosing bare root plants is really important too. So when it's possible to do that, if you're gifting a friend a plant or a tree or anything like that, just rinsing off the roots into a bucket. You can strain any part of the soil that's really bigger than like a ground mustard seed or a mustard seed rather. Just to prevent, again, those cocoons from spreading to other areas. And then you can plant as normal. if you think that you might have gotten jumping worms from a nursery in Maine, you can let them know that you think that that might be going on. And you can also reach out to Gary Fish. He's our state horticulturalist for Maine, and this is his contact information. So you can feel free to take a photo of that or reach out to me at the end if you have more questions on that. But that would be good. And lastly, you can report suspected jumping worms in Maine using our report form. So this is Maine DACF/jumping worms. Our report form is up here in the right-hand side. And that would be great if you live in the state. We are looking for areas that are not already positive on our map. So we do really want to have a good understanding on where these jumping worms are in the state. And that being said, I do also have a QR code here. You can use your cell phone, your camera app, and you can hover over it and click on the link that pops up, and that'll bring you right to our report form. And you can report jumping worm presence in the state of Maine that way as well. If you do decide to do that, please make sure you include photos of the worm. Otherwise, we aren't going to be able to identify it for you. And again, the juveniles this time of year, quite difficult to identify through photos. Hand lens is really the best, and that can be hard. So I think that's what I have for you today. I think I'm a little bit early, but I know that there's time for questions. So I want to thank you again for tuning into my presentation. This is my email and my work phone number. So please feel free to reach out if you have any additional questions. And I'm happy to answer questions here as well. So thank you.
Thanks so much, Brittany. Let me get my camera back on. Alrighty. So, anybody feel free to, I don't know if you're going to be able to unmute, but if you want to ask a question live, maybe I'll give a little bit of time for that first before I go back through. Gary and some of our speakers have been working great to answer just about every question that's come through, but I can start looking once I hear. So does anybody want to ask any questions live? You can turn on your camera and turn your mic. All right, let's see. I might have to. Nancy, you should be able to, let me know if you, there you go, perfect.
I think I can. Yeah, thanks. Brittany, I'm so curious to hear more about the kinds of sites in the forest where you are finding jumping worms. Do you think that they are mostly associated with bait travel, or like how outside of a garden or infestation via a nursery or a plant, active planting route? What are the kinds of sites where you are finding these things? Are they spreading along floodplains? I mean, I imagine they are. Yeah, I'd just like to hear more about in the natural setting, where are you finding these?
Yeah. Thank you for your question, Nancy. So in my experience with finding these jumping worms, it is most often at trailheads. So a couple years ago, I had done a lot of different kind of ground monitoring with the detection method using the ground mustard to try to figure out where worms were and where they weren't. So I really tried to prioritize things like boat launches, trailheads, composting areas. And most times when I was finding any kind of worm activity, it was at the start of a trailhead. So this could be people putting on their hiking shoes or their boots or anything with a nice tread and going through a trail, or an ATV trail could also be problematic as well. I wasn't quite finding them often at boat launches, except for that one photo that I had that was at a boat launch in Orrington, which was also a common fishing spot. And the other thing, I was finding them at a community composting area as well, and that's harder to regulate because it's just kind of open for everyone to contribute to, and it's a community source thing. So that's a bit tough, but I would say those are the top three things. Anywhere where these cocoons could get into a tire, a tread, anything like that, landscaping equipment, they can move right in. So new construction would also, I would consider it a high risk area, although I did not confirm any detections through that.
Any other live questions? Go ahead, Lee. You can unmute yourself.
Okay, hey there. I was just wondering, I had put in the chat, but I've identified this from personal experience and I think from reading, but I'd like confirmation. My understanding is that the Aminthus are semi-aquatic. And I know that because I threw some in a bucket a couple of years back and then went back to the bucket a couple days later. And I have a video of this and I show it to people and I think I have leeches in the bucket, but I don't. I have the worms. So I'm pretty sure that's the case. Whereas Lumbricius drowns. It's sort of like the Salem witch trials version. If it drowns, it's the good guys. If it doesn't, it's the jumping worms. Am I right in this understanding? Thank you.
Yeah, thanks for the question, Lee. I can't say that. That is some crazy feedback there. Yeah, I cannot say whether or not I've personally seen jumping worms coming out of a bucket like that. I've heard this soap, actually adding soap to a bucket of soapy water, if you are throwing them in, if you're hand removing them, can actually help break that surface tension and prevent them from being able to get out of the bucket.
I'll jump in on this one. I've seen a presentation, I think it was Josef before, so maybe he'll talk about it in the presentation, where they can swim, and I think they can swim a little bit upstream to move. Lee, I'm just muting you until you talk again, because for some reason there's a little feedback. So what I would guess, if I just had to make an educated guess, is that when they're in the bucket, they'll probably come to the surface to get some air occasionally, if I had to guess. So maybe what's happening, or you know, what you should always do then is add some soapy water, because like Brittany said, that breaks the surface tension. So that'll make it much more challenging for them to come to the surface. Was that the rest of your question, Lee? I just am asking because you unmuted, so I didn't know if you had anything else you wanted to ask.
Sorry, I'm having a little trouble with this application. I'm accustomed to Zoom. My point was, I may have misspoke. I wasn't suggesting that they jumped out of the bucket. I was just suggesting that they were swimming very actively, let's say, in the bucket. I think there may have been two species. I will share with all of you this 1-minute video. I'll get it to you, Hillary. I think I have your number. So I'm not saying they jumped out, but I am saying they lasted 72 hours and then I was a bit disgusted and threw them in the trash. So anyway.
Wow. Yeah, that's it's important to know. That's a little creepy.
I see Hildy has the hand raised.
Yeah. Okay. Is my mic on?
Yes, it is.
Well, I'm glad Lee asked that question because I have a little water feature in my yard, and I swear that, you know, I pull jumping worms out of this little pond. And I looked up online and saw that, you know, we do have aquatic worms in Maine, freshwater aquatic worms, but these look just like jumping worms to me with the clitellum that goes all the way around the body. And I don't know how long they're staying in there, but definitely they are, you know, in this little pond. So whether they're moving in and out or, you know, living there all the time, I don't know.
Interesting.
But they're definitely there.
Yeah, this is honestly the first time I've heard of them really thriving in a body of water. So I don't think I have enough knowledge to speak more on it. If somebody else does, I definitely would love to hear more. But we'll also compile these questions and try to get answers to the ones that we can't get to today. So that might be one we revisit after doing some research or maybe contacting people, because that's important to know. And I don't have a good answer for that. Yeah.
And thank you everybody for taking the time to ask questions, because this helps us know how to help you better.
Yes.
So,
yeah.
All right. So I see Caroline Elliott is next in line.
Hi, thanks. Sorry. I've put jumping worms, you know, hand collected them and put them in a bucket with water or vinegar. Is vinegar okay? That's one question. And then the second question was, if I'm collecting adult worms that might have eggs in their body that they haven't laid yet, does that mean I have to be careful about discarding the water from that? Would the eggs survive even as their bodies decompose around them?
Two really great questions. Thanks, Caroline. So for the first one, I have heard of people using vinegar and it being successful. However, I think that if you are hand removing a lot of the worms, you will need a lot of vinegar because you really want to make sure that you're covering the worms. So in that case, you might save some money by just using soapy water instead of using vinegar. And then that way you're not introducing vinegar into your gardens or anywhere else. So I'd say either is fine, and I've heard both are effective. As far as the other part, when you are using or when you have the adult worms that potentially maybe they are in the process of laying those cocoons, if they don't have that clitellum is their reproductive structure. So if you are working with a bunch of juvenile jumping worms or worms in general, it's less of a concern in that case, just because they don't have any capacity to produce those cocoons. If they are adult jumping worms, they do have that clitellum structure, and you're trying to kill them. There is concern in my mind, how much dirt is covering them, if you're washing them off, that kind of thing. Anytime we're working with the adult worms and we're pulling them out of soil, I would just treat any kind of soil particle as a potential cocoon. So it would be the same kind of precautions, whether or not they have fully sloughed off those developing cocoons or not. So again, straining, if you want to put them and strain out that soil, you can solarize that soil or bag that separately and just kind of bake it in the sun for a little bit, kill the worms in soapy water, and then you can throw everything away. I think that would be a good way to approach it, but I'd invite other suggestions on that too. To my knowledge, I would treat it as if it did have cocoons though, so I would be straining it.
Great. I see Phil has a hand raised. Go ahead and unmute.
Yeah, great presentation. I have a question about natural predators that could be distributed to go after the cocoon stage or the early hatchling stage. Some research that I'm doing as well. I cover all of New England and have a kind of bare ground sample trial going on, but with nematodes specifically, has any trials or has anything been executed to see efficacy of applications of nematodes targeting those two specific life stages?
Yeah, that's a great question, Phil. Thank you. I
Yeah.
can't say that I'm familiar with any research that's going on with nematodes specifically targeting the cocoons themselves. For natural predators, because jumping worms are an invasive species, as we know at this point, they didn't evolve or grow up with our native predators, and so there are things that will go for it, like birds and snakes and whatnot, that will just eat any kind of worm. But specifically for cocoons, that's really tough because again, they're so indistinguishable from the clumps of soil. I don't know of any larger critter that would be going for it specifically. Nematodes are a whole separate beast on their own. I can't say that I'm entirely familiar with nematodes. So it's possible, and I'd love to hear if there is more research going on that, because I think that has a lot of potential to be quite cool. But it would likely be from their native region where jumping worms are native, and there's biocontrol for that, and long process. So I don't know, but it could be interesting to see how that research goes.
Yeah, just real quick, I am running some trials. So I work for a company that supplies beneficial insects and biological controls. So we are doing some trials on bare ground material and as well as potted material. So if anybody, is growing and doing things like that in a container, we are releasing nematodes to target jumping worms specifically. So I can always give you and provide feedback of what I see in the trials, but from initial, applications, it's looking encouraging. Again, they're not by any means labeled for it, but that's something that we're trialing is beneficial nematodes targeting those two life stages. So early spring applications, as well as we're going to do some late summer, early fall applications.
Yeah, I'd love to hear more about that. Yeah, you have my email and my number now, so I'd love to be kept in the loop. Maybe we can work on something together. Thanks, Phil.
Perfect. Yeah, you're
Same.
welcome.
Any other live questions? Okay, seeing none. There's been a lot of activity in the chat and lots of answers and such. I
Oh,
think,
good.
oh, yeah.
Oh, no, that was me. I was just saying
sorry.
good.
So if anybody asked a question a long time ago in the chat that you really don't want to get missed, maybe send the question again now. So it's down to the bottom. But this is a fun question. Do cocoons make it through the gut of a bird who ate an adult worm? I don't know the answer. Does anybody know the answer? That's a good question.
Yeah, it is. I don't think I know enough about birds to say. Because I know that they will oftentimes eat like seeds, and then incidentally, their scat will be just spreading seeds around. So I don't know.
Oh, awesome. Erin says she wrote a paper where we looked at that and the cocoons did not survive.
Okay, well, there you go. Good to know. Thanks, Erin.
Erin, can I ask how you did that? The method was? That's pretty cool.
Yeah,
Okay,
can
cool.
you hear me?
I can.
Okay. Yeah, we caught robins in a mist net, and then we force fed them cocoons, and then we kept them in a cage overnight, like whatever length of time we had researched it would take for something to pass through their guts, and then we sorted through the droppings. So it was pretty gross. I'll put a link to the paper in the chat.
That's
Yeah, please
fascinating.
do. Thank you.
Important work, even though I'm sure it was rough. Thanks for doing it. All right, let's see. There's so many back and forth in this chat here.
Maybe one more, and then we
Yeah.
have the next presentation to load up. If you're seeing any. I'm seeing Gary is really a rock star going through and answering all of these. Thank you, Gary.
And Erin just shared that robin paper in the chat.
Awesome.
All right, I think I'll get, oh, nope, Hilda, you want to ask one more question?
Yeah, I do. I mean, it's about this whole, the annualness of these things. I mean, so I really feel like I see, adult jumping worms in my garden in the spring. So is it possible that, and I mean, if if it's old age that they die of, that if they're hatch late in the year, that you could have an adult in the spring?
So
I mean, if it's an old age kind of thing.
Yeah, so they die as adults, typically the first few frosts, and that's because again they can't burrow down to really save themselves from the harsh weather, and even in like the most prime conditions, they still only lived for about a year and a half. So they're just not, their biology isn't for them to live very long. As far as you seeing potentially adult jumping worms in the springtime, I'd say that it's certainly possible, because when we're looking at the annual lifespan of these guys, they are emerging in that early May time. If they eat fast enough, their whole goal is to just eat a whole bunch, grow a whole bunch, and then become of that reproductive age. It's not necessarily that they aren't present in that May, that June time. It's just that the adults are present in such small percentages that more often than not, the photos that we receive are of the juvenile worms for that time period. So you could be seeing them. And again, you know, if you wanted to send photos of these potential worms with the clitellums, that would be a good way for us to identify, or now you have the knowledge to identify them yourself. So you could do that too, but I'd say it's possible. Yeah.
Okay, thanks.
Yeah, thank you.
Great. So Dr. Josef Gorres is a professor of ecological soil management at the University of Vermont and one of the co-directors of the Healthy Soil Collaborative, which investigates jumping worm ecology, their impact, and distribution. His research includes work on soil fertility, restoration, erosion, and earthworms. All right, so.
Okay.
Professor in ecological soil management.
Cool.
I work at the Department of Agriculture, Landscape and Environment at the University of Vermont. We used to be called Plant and Soil Science. And I'm also here as the co-director of the Healthy Soil Collaborative. I'm talking today about organisms, heat and chemicals to control earthworms. And the main organism we're going to talk about today is the jumping worm, but also humans. I'm currently in Newfoundland, and I'm probably staring at something like that while you're listening to my talk. I'm sorry that I'm not with you in person. So one thing that I'd like to say right now is that you should note your questions in the chat of this meeting, and I will then answer those questions when I get back from vacation. Well, number one organism are humans, Homo sapiens, and we have a lot to do with this invasion, and we also have the power to maybe do something about that invasion. So humans are the cause of the invasion, but humans can also be part of the solution of this invasion, right? So this is really important to realize that, yeah, we're causing this by having moving earthworms around with horticultural goods, or having them in our garden, or many other ways of moving them around. But we can also be part of the solution by doing the right thing. You must wonder about the logo, the one up here that is sort of a circle with soil and a plant growing out of it. It's the logo of the Healthy Soil Collaborative, and we are a group of scientists and practitioners who are interested in controlling the invasion of jumping worms. So we are hoping to work together to stop invasive jumping worms and save our ecosystem, gardens and forests. And that's really important. One of the things that we want to do with this is to solicit donations through a fiscal sponsor called Ecologistics, and then give that money to researchers who have good ideas. We like to take an integrated approach to this problem. So we want to learn about the spread of jumping worms, their impact, their control, and how to restore areas that have been impacted. All of these 4 areas of research are very interrelated. So the spread is related to the impact, because wherever these worms spread, they will have an impact on water quality, soil quality, maybe plant life, etc. Control is related to that, because we can control them, we can reduce the impact that they have. And then finally, restoration, of course, is related to all of that. We try to improve soil quality by restoration, and we would have to have controlled these earthworms in those areas. So if you have a soil that has been impacted by jumping worms, then likely the plants that will be able to grow there will be different to the plants that were grown there before. So we need to take into account that little snippet of information, and maybe think about restoration in terms of, well, let's first control the earthworms, and then secondly, how can we bring back the function of that ecosystem rather than the individual exact plants. So our detailed plan is integrated multidisciplinary science, and it will involve a lot of education, training, outreach, and policy to bring about change in the way we look at jumping worms. And so the research areas we are looking at is spread, impact, control, restoration, as I said before. And within each one of those, there's different things that we might want to look at. So for example, from spread, what is the mode of transport? What moves them? We know what flowing water does, we know what horticulture does. Are there any others? How does behavior of humans change the spread? And how does climate have an effect on the spread? Then we have to acknowledge that there's psychological distress when you find jumping worms in your own garden. And we need to address that as well. So right now, I would say take a deep breath and relax a little bit. So these worms are obviously, you look at Facebook pages, they're obviously a menace to many people. And so I want to acknowledge that, but do breathe and relax a little bit about them. There are different things that can be done in the future. And even now, you may be able to lower the impact of these worms. And the impact, we're really interested in water quality, greenhouse gases, biodiversity. We want to control them. We need to look at pesticides. We need to look at biocontrol and maybe some cultural measures, maybe heat as a way of controlling them. And some of that will require that we make pesticides and biocontrol agents legal by having them certified and registered by the EPA and by the state departments of ag. And then we can think about environmental management as well. And then finally, there's restoration. So as much as horticulture is at the root of some of this, they can also be part of the solution by maybe producing native plants that can withstand the changes that have occurred in ecosystems inflicted by jumping worms. So for that, we have assembled a team of experts, soil scientists, ecosystem scientists, outreach specialists, forest scientists, horticulturalists, science communicators, landscape practitioners, biogeochemists, and toxicologists. And we have probably, as a group, we probably have many more capabilities that are not listed there, but we're a group of 15 scientists from 10 institutions in the USA and Canada. And we have the same aim, even though we might be doing research on very different things. Why bother? Well, there's the broad impacts of earthworm invasion. So not just jumping worms, but other earthworm invasions as well. And the immediate impact is on soil structure, and that will change how nutrients are cycled. And you hear a little bit about that from Justin Richardson tomorrow. Anyway, so those cycles will alter the microbial processes and the soil food web, and that will have an effect on plant community, the habitat for vertebrate species. And all of that together will eventually have some kind of societal impact that will cost us. So that might change disease dynamics. Just the removal of the organic layer might change how the Lyme tick is spreading, right? And that might be an impact. It facilitates invasive species and weedy species. Wildlife habitat is going to be changed. Soil water quality is going to be changed. Water quality, drinking water quality, maybe in reservoirs, New York City reservoirs that are protected by big forests around them, in the belief that the forests are sinks of net uptakers of pollution rather than sources of pollution. Climate change but even things like what about wind erosion and fire, right? So soils that are invaded tend to be drier, and so they are more likely to cause fires in forested areas. So what you saw there is basically a big concept of, there's going to be damage, there's going to be problems. How about the cost of that damage, right? So here's a graph showing showing how the cost of storm damage, biological invasions, insect infestations has increased from 1980 to 2020. So this is the total cost over these 40 years. And storms it's about $2 trillion in storm damage over those 40 years, and about 1.1 or 1.2 trillion dollars in damage done by biological invasions. So it's of the same order of magnitude, and it's disturbing how much this costs. Right. So if you look at how damage cost has increased for storms, for looking at the period from 1980 to 1999, and compare that to 2000 to 2019, then storm damage has increased by 400%, so fourfold, whereas biological invasions have increased by 15-fold. That's huge. So these invasions are becoming more and more common and more costly over time, and seem to be outstripping even storm damage increases. So these graphs were for damage costs. They were not for cost that we have to spend on controlling the invasions, right? So generally for invasions, we have this invasion control cost curve. And basically what you have on there is, the more there's on the y-axis, you have the area occupied by the invasive creature. And then on the x-axis is sort of the economic returns on the dollar spent on various measures. Right. And then there's a curve that looks like an S curve, starts over here and then increases to a point where maybe the cost flattens out a little bit. The cheapest way of controlling an invasion is to prevent it, right? So for every dollar you spend in prevention, you save yourself $100. The next most important one is, so once species arrive, you may want to think about quickly eradicating those species while you can. And there the cost ratio is a little bit worse. So $1 will get you $25 in savings for containment. So you get more area infested, and then you start thinking about containment of these worms, in this case worms, right? So you try to keep them where they are and trying to stop them from moving around. And there the cost ratio is $1 spent will get you like $5 saved. And as that invasion continues, then for every dollar you spend, you only save $1 in economic returns. So that is the cost curve. So that adds to the damage cost of this. So having said that, invasive species just don't have it easy, right? So it's not that easy to come from one place on the planet to another place on the planet and get established. And the very first thing that needs to happen is you need to get there. Right? So the transport is important. For jumping worms, they had to travel the sea, right? They had to travel across the ocean from Japan and Korea all the way over to North America. And so that transport is, that ocean is a barrier to transport, and something has to happen in order for plants that might carry the cocoons or something like that to get across the ocean. And so this is as much a problem as it is an opportunity to disrupt the invasion, right? So, for example, good phytohygienic measures might solve the problem, right? So you say only bare root plants can be imported. So that would then be the way to prevent the erosion. Prevent the invasion. The next step in this thing is introduction, right? So we're able to transport the invasive earthworm across the ocean, and now it has to be introduced somewhere. And oftentimes, invasive species are introduced into things like botanical gardens, where they live in captivity. They're well looked after. They may not be ready to jump into the wild until maybe the climate or the soil has changed to a point where organisms can escape. And yet again, here you might want to take some preventive measures, or you might even say, well, we never really thought of bringing these worms in. Let's eradicate them in captivity before they can get out. So for each one of these steps, there is probably an opportunity to stop the erosion. That's in the invasion. Where is the invasion right now? This is a map from Moore et al. 2018. It shows the northeastern part of North America, and there's gray areas that are light in color and gray areas that are dark in color. The light areas is where these jumping worms cannot exist at this moment in time because of climatic limitations. The dark areas is where they can exist. They may not be there, but they can exist based on climate. The northern limit of the spread in the northeastern part of North America is this black line. So it goes from Halifax through Fredericton, through Toronto, into Niagara Falls. So that's basically the northern limit of this, and maybe you might have some up here in northern Maine. But anyway, so that's currently the northern limit of the spread. So why is it important to know the current distribution? Well, if you know what the current distribution is, you can go back to the sites where these earthworms exist, and you can then look at the environmental conditions under which they exist, right? So what is the vegetation, what is the soil type, what is the climate, and so on. And you can make a relationship between that and the jumping worm presence. And from that, you can figure out what are the areas that are at risk of invasion. And then you can target those areas with preventative measures and controls, so that they are protected. And you could, with this distribution, you could also figure out what's the impact of these worms on natural resources and ecosystem services. And finally, you can design better restoration and mitigation strategies. So that distribution is really important. So Homo sapiens is not really good at identifying jumping worms. Every March, I get calls and I get images sent to me that show earthworms, and they say, I've got jumping worms. It's horrible. Look at this picture. And most of the time, you're not talking about jumping worms. You know, the images are of European earthworms, also invasive creatures, but not jumping worms. And if I see a picture that was taken in March, May, and May, I usually am very skeptical that these are jumping worms anyway, because at that point in time, you just have juveniles. You don't even see very much of the characters that would allow you to identify these worms as jumping worms. But in March, there's usually a huge event like a big rainstorm, and suddenly you have a ton of worms come out of the ground, and then you go, OMG, I've got jumping worms. Most likely not if that happens in March and April. So you're not going to see clitellated worms, so adult worms, before mid-June in gardens, and July at the earliest in forests. Anything you see before then is unlikely to be a jumping worm. This image shows really clearly how a European earthworm differs from a jumping worm. European earthworms have clitellums, and those things are or saddles. So these go only 3/4 of the way around the earthworm. They tend to be raised, and they seem to be similar in color to the body. And then for jumping worms, you have a milky kind of color of the clitellum. It's smooth. It's pretty much flush with the rest of the body, and it goes all the way around to that earthworm, right? So that's the most distinctive difference between these worms. Another distinction, and this goes for some of the juveniles as well, is that they seem to have something that looks like a tongue, right? So sometimes you see this protrusion come out of the mouth, and so that is another sign that doesn't happen with European earthworms. Another way to distinguish between lumbricids, so European earthworms, and jumping worms, is that they have a different kind of arrangement of setae or bristles around each segment, right? So jumping worms have lots of bristles all the way around each segment, whereas the European worms have just a few. If you look at the worm under the microscope or with a good hand lens, you might see something like this. You see lots of little stipples on the ridge of the segment, and so that would indicate that you have a jumping worm, and those are already present in juveniles. These are the three most common species in the northeastern US. Metaphyll heggendorfi, Amynthas agrestis, and Amynthas tokiensis. They differ in size, and that's pretty much it. Metaphyll heggendorfi, when they're adults, they actually have two little dots at the just ahead of the clitellum that you can see. Actually, you can see pretty well here. There's one dot, another dot. That's the only way to distinguish between the three of them. Tokiensis and agrestis look very similar. One is more slender than the other, but in reality, you don't want to go by that kind of shady comparison. Identifying juveniles, you can't really do it, but there's some clues that would allow you to identify the juveniles, at least to jumping worm, right? Not to species. And so one of these characters is their behavior. Like, so here we go. And most of you probably have seen this kind of behavior. Jumping worms don't jump, they wriggle like crazy like that. And there's only one other worm maybe in the northeast that might do something similar, and that would be the red wiggler. So wigglers do similar things. They're maybe not as vigorous as that bouncing around in your hand, but they wriggle quite a bit. And the trick is to understand that they look different, right? So if your worm has these stripes, when they stretch, these yellow stripes between each segment, then you have a red wiggler. Also, you can look for a yellow tail at the end. That yellow tail also indicates that it's a wiggler of some sort. So first have a look whether your worm is doing that wriggling in your hand, and then secondly, then look at whether you see any of these stripes. Red wigglers are also known as yellowtail or as tiger worms. So you just remember that for that. So you can sort of identify juveniles in this fashion. So the other thing that's important is to know a little bit about the game of life for these jumping worms. What's this life cycle? Questions like, when do jumping worms hatch? When do they become adults? How many cocoons do they produce? And those are really important questions for understanding the biology, but also for understanding when you might want to apply some kind of pesticide, if there was one, and when to maybe collect them or when to do something else to the soil in order to exclude these worms. So the average Wisconsin jumping worm, actually the same species we have here, you see juveniles in May and June, and then in July you start seeing some adults. August there's more. September and October you probably have mostly adults in the mix, and then from November to April the worms exist as cocoons. So this is a very simplified version of this life cycle, because the cocoons also exist all the way through the year. It's not just these 6 months, it's 12 months of the year. And so we are dealing with a cocoon bank, and I'll say a little bit more about that in a minute. So one other thing to recognize, that adults emerge at different times of the year, from year to year, right? So there's a pretty wide span. Here's 4 years of jumping worm data from a forest, and the curves all look very different. And one thing that is even more surprising is that the adults in these 4 years emerge over the first adults, not all the adults, but the first adults, the very first time you see an adult varies from sometime beginning of July to about the end of August. So it can be as different as that, depending on the weather, temperature, and moisture. And then as you go into September and October, you start seeing mostly adults. And after that, sometime at the end of August, actually, there's a die-off because there's the first frost, and then the die-off continues as frosts become more frequent. And by the time you get into December, most of these worms are dead. They're truly annual worms. Adults produce about 0.5 cocoons per day. That's, I think, important, interesting figure. And hatching seems to occur even in parts of the winter. And we'll get to the hatching behavior also in a minute. So there's a huge interannual variation in the population levels of these worms. And here's 15 years of data. There's a couple of years missing. I haven't entered the data yet. But you see that the peaks are very different. There's lower peaks around 100, and there's some that are around 200. For 2019, which is not shown here as this missing data, it was more like 300 per square meter at the peak. And so what causes that is a really good question. Is it something that we do, or is it just a variation in the weather that causes these variations in the population levels? But these variations are important because you may want to know when you want to apply a pesticide, right? So when is the pesticide most efficacious? When you go back here to the individual years, it might be most efficacious to do this when the juveniles have a peak, right? Or definitely before the adults emerge. Juveniles tend to be more susceptible to pesticides. And I'm saying pesticides, so you can't really legally use pesticides on these worms, right? So you may want to say that, right? So it's important that we get some information on the timing, because that goes a long way to maybe protecting other organisms like ourselves, or non-target organisms that are in the soil, like pollinators, right? So you may want to do this before the pollinators get really active. Really important to know more about the timing. So heat is one way to control jumping worms. Ideally, you want to control both the worms and the cocoons at the same time. So Vera Krischik is going to talk about controlling jumping worms at length later tomorrow, the last talk, and you want to definitely listen into that. So I will concentrate on controlling cocoons. By the way, these are 5 different stages of cocoons. The important ones are ready to hatch, stage 5, segmented and pigmented worms, embryos in the cocoons, and then there's one that we call arrested development, stage 4. Segmentation is easily seen. The body has extended quite a bit from stage 3. Where it's like a lighter blob and darker blob in stage 2, and then just albumin in the egg casing, the cocoon at stage 1. But these 2 are kind of really important because they are the ones that might hatch the following year if there's any of those left in the population. So Dr. Nourian has done an experiment where her hypothesis was hatching is regulated by a genetic timer. And so she tried to figure out, is there a timer at all? So is time of year a factor in the hatching behavior? So she collected thousands of cocoons. She incubated them at different temperatures, 5, 10, 15, 20, 25, 30, 35, 40 degrees Celsius, to see whether there's an effect of temperature as well. She incubated for 260 days, and she had 150 cocoons per temperature treatment. Incubation was in the dark, in DI water, and at constant temperature to exclude any kind of stimulus that might occur because of light variations or chemistry variations, or because there's temperature variations. She looked at the embryo stage once a month, and she looked at hatchlings twice a week. So she counted hatchlings twice a week in petri dishes at these temperatures. And so is there hatching timer? So I would say, and Mariam would also say, yes, there is a hatching timer. If you look at this graph, time on the x-axis and number of cocoons hatched. Percent of 150 cocoons hatched. Before February, no hatching. Hatching started for a 5-degree cocoon in sometime in February. Then at the end of February, 2 days later, you had cocoons that were held at 10 degrees Celsius started to hatch. It's 50 degrees Fahrenheit. And then within a month, 70% of the cocoons in those petri dishes held at 10 degrees had hatched. So it is both the timing issue, sometime before or close to spring equinox, and it's a temperature issue as well. And these are similar results to Blackmon et al. 2019, who worked on Amynthas agrestis, so different species, but they had 120 days before the hatching started at 10 degrees, just like with us. That turned out to be the end of February and beginning of March. So they weren't really interested in timers. They were interested in how long does it take, right? So, but this is really, really cool, that there is a timer. And so again, this could lead into some kind of way of controlling these earthworms. So 10 degrees Celsius seems to be optimal hatching temperature. How did cocoons develop? So if you looked at what was left after 260 days of incubation, then you saw this. The different temperatures of incubation on the x-axis, and then percent of the cocoon status on the y-axis. So hatched is this darker color blue, undeveloped. That means they were like stage 1 through stage 1. The orange, arrested, stage 4 is the green. Ready to hatch is the light blue, and the dead cocoons is the purple. And so as you get to 20 degrees, you start seeing more and more mortality of embryos in the cocoons. But at 20 degrees and 25 degrees, you also had a lot of arrested development. These cocoons are no longer developing into ready to hatch. They're holding back. And then for anywhere between 5 and 10 degrees, there's quite a few ready to hatch cocoons that didn't hatch. And the hatch cocoons seem to be mainly in the 10 degree Celsius treatment. And everything that is undeveloped, arrested, or ready to hatch might be part of the cocoon bank where cocoons might hatch the next year or the year after that. So the cocoons are heat sensitive. We know that from Johnson and Herrick that showed that 38.1 degrees Celsius is a kill temperature for jumping worm embryos in cocoons, and that agrees with Nuri Ein's data. So that seems to be a pretty good number. Field hatching at ambient variable temperatures produce different kinds of hatching behavior. It is no longer confined to just 30 days from end of February to the end of March, but it's spread over a longer period of time. So if you look at the juveniles per meter squared, they increase from April, when the first ones are detected, all the way to July. And then the numbers go down because they develop into adults. But all the way to July, there is some hatching. And that has to do with the fact that the temperature of the soil is not always at 10 degrees, even though the temperature may be even though the average temperature is greater than or less than 10 degrees, there is some variation. And so in April, you get a few days where it's 10 degrees. In June, you also get a few days where you still have 10 degrees weather, and then that leads to the hatching of these worms. That's sort of the theory. So basically, this develops a concept of hatching and cocoon development that leads to maybe a model that can predict eventually what next year's next year's output of these worms is, right? So how many worms do you get the next year? And I think that's really important because you want to be able to predict what next year's hatching rate is, so you can target maybe a particular time of the year, the following year. So one way the way I think about preventing transmission is this: you've got to deal with like horticultural materials. That's, I think, really important. And there's other things like hiking that will also transmit these worms to the wild. And you know, the National Park Service in some areas has boot cleaning things like these brushes that you can use to clean your boots before you go on the property. That is not necessarily done for jumping worms, but it would also help with jumping worm stuff. So the tread of hiking boots can take cocoons from one place to another, and then you might get an infestation from that. But horticultural materials are really the big ones, right? So so potted plants, compost, mulches, those can all contain jumping worms in different numbers. One should think about how can you treat these. So we know that heat is a really good killer of jumping worms, cocoons, and the worms themselves. And so you could actually use pots and compost and mulches in a solarizing sort of place in order to kill the cocoons and the worms, for that matter. But how do you do that, right? So that's of course the question. So we know that, just to give you an idea, that you know, if you look at earthworms, so here's a table from Richardson et al. 2009, Southeastern Naturalist, and it shows the temperatures at which worms do not survive. So at -5, they didn't survive. At 5 degrees, they didn't survive. At 35 degrees, they didn't survive. So worms have a very similar kill temperature as the cocoon. So we can actually probably kill both of those cocoons and earthworms in one go if we find ways of solarizing to the right temperature, so around 40 degrees Celsius. And you might say, doesn't compost get really hot? Yes, if it's done well. So here's the anatomy of a compost pile. So usually you have something like a layer on the surface that is less than, over time, this layer develops into temperatures less than 40 degrees. And then you have an area in this pile that has temperatures greater than 40 degrees. So it's basically this ochre, and then the red is also greater than 40 degrees. So all of this together will kill quite a few worms and quite a few cocoons, but not this, but not in this surface layer of this pile. But if it's done well, then you end up for a short period of time with areas that are greater than 40 degrees anywhere in this pile. And that's what you're aiming for. Unfortunately, there are some places that don't have a really good control on this. So some municipal composters might not reach that temperature. Community composters might not reach that temperature everywhere in a pile. So that's the problem. But even if you are at a compost facility that does reach that temperature, you might still get re-inoculation of the compost in the curing, in the maturation area, so where the compost is cooling down. You get that migration of earthworms from the feedstock storage into the compost curing area, and then you have contaminated compost. But even more likely scenario is that some contamination occurs in areas where resale places are storing so garden centers, for example, where they're storing their compost for resale. Anyway, so you can solarize these horticultural materials, and you do that by creating these packages, and you can do this at a much bigger scale. But this package is basically 6 mil painters plastic sheeting all around, so wrapped up all the way around. And at the bottom of this, there's a layer of cardboard. And I can't remember whether this was cardboard or whether this was that 3/4 inch styrofoam pad that is used as insulation in roofs and stuff. But anything that feels warm to the touch is probably a good insulator. You've got to insulate the bottom of this because the bottom is influenced by heat loss from the solarized package into the soil, and that means that the bottom of this pile stays cooler than the 40 degrees. It takes a while to reach that 40 degrees over the entire thing. So if you look at these curves, look at the bottom curve, this dark blue curve. That is the curve, the temperature curve for what is at for the lowest layer. So 8 inches, this is the 8-inch package. So at 8 inches down, this is the curve that you see for temperature over time. And you've got to reach that 40 degrees or 39 degrees or whatever it is for a couple of hours. And it took 4 days to reach that. By the way, the numbers up here, the temperatures up here are temperatures that are ambient maximum temperatures for the days that we are solarizing. So it takes about 3 to 4 days to reach temperature everywhere. So you want to look at the weather forecast and check whether there's 3 or 4 days of really warm weather there. The more styrofoam or the more insulation you can give it at the bottom, the better. One of the people that I'm working with, Gardner, has converted a frame for growing plants in the springtime to a solarization thing. So he basically took insulation and insulated that frame with that insulation, and he reaches like temperatures of 60 degrees Celsius in that cold frame. So there's different ways of solarizing, and it depends on the amount of material that you need, what you want to do. You can solarize fields and flower beds, by the way. It's not as effective as sterilizing compost in wrapped up packages, but it does reach to 3 centimeters down, and that will probably reduce the number of earthworms that will emerge in the springtime. But it depends on moisture and the ambient temperature, so you want as dry a soil as possible in order to allow that heat transmission to reach deeper. So quick thing on chemicals and organisms. So pesticides, no pesticides certified for earthworms, period, at this point in time. So there people are working on that, and just wait for Vera Krischik's talk. There's predators. So a lot of people ask me, is it safe for chicken to eat these worms, because these worms are bioaccumulators of toxic trace metals, and it is safe when your soil is clean of metal pollution. So let the chicken go at it if your soil tests from the University of Maine or anywhere else comes back clean. So it has to be a special soil test that includes metals. It's not, it should not, it can't, the typical fertility soil test doesn't tell you much about the toxic trace metals. So you want to have a specialized test that is offered by the University of Maine and other universities, land-grant universities around the nation. We are important institutions for farmers and for homeowners, even just for that, soil tests. I'm glad that other chicken moved so fast, because we would have missed the slurping of the worm by this hen. So as far as pesticides, they should control both worms and cocoons, and that's really difficult. So most tests are done on the worms and not so much on the cocoons. Cocoons are the cryptic side. We don't see them very often. They're survival structures. They're very hardy. They form multi-year cocoon banks, and we need some experiments that look at whether pesticides can increase cocoon mortality. Entomopathogenic fungi. So in our lab, we Mariam Nourian, who's now actually an assistant research professor. Congratulations, Mariam. She's worked with Boveria bassiana, or Botanigard, and she worked with a granulated formulation where she grew the fungus on millet, and then she put the millet into X number of grams of millet into pots that were in potted plant pots that were infested with jumping worms. And she got something like 75 to 80% mortality rates for that formulation. Worked really well. So once again, this is not certified for use on earthworms. Pesticide chemicals and pesticide organisms are not registered. So Botanigard actually works for that formulation, but again, it's not on the label that you can use them for earthworms. So we have whiteflies, aphids, thrips, psyllids, mealybugs, leafhoppers, and so on, but no mention to earthworms. And it says listed food crops, forestry, orchard crops. That's where you can use them, right? So you need to read the label, and there's more to the label than just that, right? So, for example, it tells you where to apply, when to apply, crops and plants, how to apply as a powder or as a suspension, a drench, what is allowed. And it gives you, and this is to protect yourself, but also non-target organisms. This is not something that is entirely trivial. Saying you can't drink this stuff, right? So, and it will tell you that on the label. But here it tells you a little bit how to apply it. So the second page or page 9 of 13 on the label. And then there's also information about which crops and ornamental organisms you can use these that you can use Botanigard on. So there's a lot more information on there. You should read the label. It's very informative and helpful. But again, at the moment, even Botanigard is not certified. I'm not a certified pesticide applicator, so I cannot give you any advice. Other than that, the label is the law. Read the label, and anything you use with intent to control a pest is a pesticide. And so those are important things to remember. So in conclusion, I would say that researchers are working together in an integrated approach where we provide more education and more research to solve the problem. So there is hope for us researchers because we want to solve the problem, and for you because you also want that problem solved. But at the moment, we only have part of this picture, and we really need to do more research. But I let you go by saying there is hope, and I will continue exploring Newfoundland. Important, please write down your questions. Either submit them to the organizers of the conference or in the chat box in the chat, and I will answer those questions when I come back from Newfoundland. Thank you for your patience.
It says that you should clean your boots before you go hiking, but you know, do you clean them at what point? Because you don't want to take bad soil into the trail, but you don't want to bring it home either. So I'm not sure how you would do that at home.
Ideally, you do it before and after.
And so what do you do with the soil?
If there's a way to contain it and put it in the trash, then that would be okay. Put it into a plastic bag.
The trash that goes to the dump, you mean?
Yeah.
Okay.
Yeah, and you could like brush your boots off into a trash container. Yeah, just don't move it somewhere else. I mean, that's the That's what you don't want to do. Try to leave anything where you are, if you have to.
I also want to mention too, Erin just threw a really great link in the chat, Play
Yep.
Clean Go. If you click on that link, they actually have an interactive map that contains boot brush stations at trailheads and other places where we are really trying to slow the spread of these invasive species, not just jumping worms, but also invasive plants too. So that's a really great website to check out as well. Thank you, Erin, for putting that in the chat. So thanks everyone again for such a nice conversation in the chat and otherwise. Hopefully people are finding things helpful. So our next presentation here is Erin Buchholz, and she has been the plant health specialist at the University of Minnesota Landscape Arboretum since 2018. She focuses on finding the safest and most effective management methods for plant pests while helping to change public perception about wildlife impacts on plant communities. Without further ado, Erin, I will give you the microphone.
Awesome. Thanks, Brittany, and thank you, Hillary and Gary, for putting this on. I've been excited about this for a while. So yeah, I was asked to speak about plant sales and then some forest field trials that we had been doing for the last few years, and I don't have a ton of time, so I'm going to go really fast. Here are our learning objectives. I will say I will also put a link to the PDF of this in the chat after I'm done with this. So the links that are in my presentation will be clickable in that PDF. Feel free to download the PDF if you want to. Otherwise, I'll leave it to Hillary and team to put them in. But these are just our learning objectives. Since we're going through it, I'm not going to read them. A brief history of the Minnesota Landscape Arboretum and the Horticultural Research Center. They are both located west of Minneapolis, about 40 minutes drive west. And I began this job in July 2018. And within 2 or 3 months of starting the job, we got a message from our Department of Natural Resources saying, we hear that you have jumping worms on your site. Can you go out and verify that for us? I had been teaching in public schools for the previous 5 years, so I was a bit rusty with horticulture, and I certainly had never heard of jumping worms. So I needed to download as many fact sheets as I possibly could, learn about them, and then I went and scouted 22 sites across our 1,200 acres, and I found jumping worms in 13 of those 22. The next year, an even higher percentage. So we had jumping worms everywhere, and we still do. Well, not everywhere, but in many, many places across our landscapes. We have wetlands, we have forests, we have prairies, so it really depends on the actual habitat and how well worms do in those areas. So we also confirm jumping worms at our Horticultural Research Center, which is where we research plants for winter hardiness or breed them for flower color or other characteristics. So we have a production nursery and greenhouses over there. And yes, a lot of our nursery areas and even our wood chipped beds where we plant out the research plants have jumping worms in them too. We have anecdotal stories from our horticulturists saying, I saw them in the early 2000s, others saying they saw them far as back as the 1980s. So we know we've had them for decades. We just never really paid attention to them because even when I went to college at the University of Minnesota in the 1990s, all of my professors said, earthworms are good. Earthworms are good for soil. If you have earthworms, you have healthy soil. So obviously we're rethinking those kind of things. So how did they get here? Of course, this helps us with making management decisions today. It's important to note that the United States Department of Agricultural, APHIS, which is Animal Plant Health Inspection Services, used to inspect a minimum of 2% of all plant consignments that were presented. They have since changed that. There are certain plant consignments now that are exempt from inspections altogether, and they kind of rate it based on what risk they pose. So not that many plants that are imported into the United States are actually inspected for plant pests hitching a ride, which is kind of scary. And so it's really up to humanity, as Josef was saying, to make sure that we stop importing invasive pests or potential problems as they come in. We could really be more proactive about that. And as APHIS is getting some staff reductions happening right now, some funding cut, I'm sure you can just imagine what impacts that could potentially have. We suspect that the Arboretum probably purchased some plants that were containerized with potting mix, sort of nursery mix that was infested with jumping worms. That's our best guess. From there, we know that they did end up in our wood chip pile, which was a huge, huge wood chip pile that we just kept adding to and never actually cleaned out or left dormant for several years. And so that wood chip pile was used for decades and was contaminated. So anywhere that we put our own wood chips from that pile, we ended up infesting with jumping worms. And so we can find old wood chipped areas where the wood chips are almost completely disintegrated or eaten down that still have jumping worms. And then we have new areas too, where we've seen some jumping worms as well. We have since sold that property. We did tell the new owners, yes, there are jumping worms there. And we now are more thoughtful with how our wood chips are stored. We make sure that we store them far away from any infested areas first, and then we make sure that we use them up within a few days if possible so that they don't have time to become infested. So yes, like I said, we use this information to help prevent the spread of jumping worms. We think about the ways that they are spread. We look at our containers now when we get new stuff in. Are there jumping worms in it? If so, we contact the seller. We haven't had that happen. But then we also want to think about where we are storing our nursery mix and if it could potentially become infested. Can worms move in and out of the piles? What conditions do you need that in? Typically cloudy, rainy days make it very, very easy for them to move quite far. And then what should we do when a mulch or media pile is infested? And also, since we have infested beds already at the Minnesota Landscape Arboretum, if we have an infested pile, should we just use it on this already infested site? Yeah, sometimes we do. It's not that we're trying to increase our numbers. It's just, it's already infested. It's not going to get that much worse potentially. And since we're not allowing that to go, yeah, it can happen. I see Leslie asked, how far is quite far? We've put wood chip piles like a good 6 feet away on asphalt away from like a wood chipped bed, and they have no problem, getting that far within a day or so. So if they can move 5 or 6 feet across asphalt, in the same day, then they can potentially move even further if the conditions are right, like it's wet and it's cool enough for them to do so. All right. I'm going to talk about our state regulations. This is going to be very different than all of you in Maine. And so I want to talk about this just because it threw us for a loop. It made us have to adjust rather quickly. And since this could happen to anyone else in any other states or provinces, we just want to give you the heads up based on what we've experienced here. We've had a couple different recent changes, and by recent, I mean in the last year and a half. So as of last July, less than 1 year ago now, Minnesota Department of Natural Resources has added jumping worms to the prohibited invasive species list, which means, according to the DNR, it is a misdemeanor to possess, import, purchase, transport, or introduce jumping worms without a permit. Notice how they did not say sell, because the DNR's intent was not to shut down nurseries or garden centers that might be infested. That was never their intention with this. Their intention was to stop the spread because it's only in certain parts of Minnesota. It is not up toward northern Minnesota yet, not showing up on any of the distribution maps. And we have looked. We've had people look all around northern Minnesota. They don't appear to be there, and the DNR wants to make sure they don't end up there. So they implemented this rule, this regulation, this law now to prevent fishing bait, essentially, from being to help that be a spread. They wanted to close that loophole. Now, nurseries and garden centers and any sort of organization that holds plant sales, those are regulated by our Department of Agriculture. So those are two separate agencies. And so whereas the DNR has them listed as a prohibited invasive species, Minnesota Department of Agriculture has not. Cool. What does that mean? Well, I'll tell you. One extra tidbit that happened this year, and this was brand new for the calendar year, the Department of Agriculture decided that if you hold a plant sale in Minnesota, you must have a Minnesota live plant dealer certificate. And what that means is an inspector is going to come out and make sure that you are selling certified pest-free plant materials. Cool. Again, what about jumping worms? They are not regulated by the Minnesota Department of Agriculture, but our Department of Agriculture will still say, even if the pest is not regulated, we don't want you selling something that could end up being a problem. So we insist that you find a way to clean it up or don't sell it. And so, yeah, there's Gary's in the chat too, talking about regulations for Maine, and that is very, very important. Know the difference between your state and province and others, because while this is helping us in Minnesota come up with best management practices and different things to think about, it's going to be different for you. So for us, our Department of Agriculture works very, very closely with our nursery sellers. They don't want to shut anyone down. That is never their intention, because if they shut us down, there's no reason for them to exist. They exist to help us and to help regulate us. And so it is very much a partnership. Our inspector was very, very kind about it. We told him exactly the types of practices that we're doing to prevent spread in our plant sales. And he was very, very appreciative and receptive to what we were doing. He also understood that our intent is not to spread jumping worms, and we will do everything that we can to try and prevent that from happening. So we hold our plant sale Mother's Day weekend every year at the Minnesota Landscape Arboretum. This year was our 57th annual sale. And a lot of our plants are produced at, like I mentioned, the already infested Horticultural Research Center nursery spaces. Well, crud, we can't sell jumping worm infested plant materials, so what do? How do we prevent worms from entering those containers? And how do we look at the current plant materials that we have to make sure we're not selling anything that could be infested? Also, how do we communicate with our customers about our practices? And so what we want to do, we want to make sure we are responsible. All of our nursery container plants for our plant sale, once we discovered that infestation, we took them out of the pots, we inspected them, we ripped them down to their bare roots, and we kind of basically made them bare root plants for a little bit. We made sure that there was absolutely nothing on those roots that could be larger than, say, a grain of sand, because as you've seen, Tokyoensis are typically the smallest of the jumping worm cocoons, and those are still about 2 millimeters in diameter. So if it was larger than a grain of sand, we didn't want it on there, and we were very, very thorough. We then found clean, bagged, heat-treated potting mix, potted that back up, and then we made sure that we did not put any of our plant material on known infested sites. We made sure that any sites that we were relocating them to were not infested. We put in pallets, we put in tables, we put down nursery cloth. We did everything that we could to make sure that we were not potentially spreading them. And I'm very happy to say that this year we've gone back and we've scouted a lot of our nursery sites, and they are looking so much better, if not, as good as we wanted them to. And before the plant sale this year, I inspected our plants, ripped a bunch of samples from each of them apart. I did find jumping worm castings in small pots of our meadowlark forsythia. Those are the only ones where I found any jumping worm castings. So I was looking, I did find some. We destroyed those plants. They did not end up in our sale. And we communicated that to the Department of Agriculture, and they were perfectly satisfied with what we did. So, yes, thank you for throwing that in too. What do other people do? So in Minnesota, we have some best management practices that were developed with the Minnesota Nursery and Landscape Association, but really I look to a guy named Nick Sargent. He has a company called Sargent's Gardens in Minnesota. He found jumping worms on his sites. He has a couple different retail and production sites, and he wrote an article for the Minnesota Nursery and Landscape Association's trade magazine, telling other business owners in Minnesota, we have jumping worms, don't be afraid, here's what we did, here's how it worked. They hired a marketing guru to reach out to their customers and tell them educate them on jumping worms, tell them, hey, we have them, but we're not going to share them with you, and here's how we're going to make sure we don't share them with you. They surveyed all of their customers, and it was unanimous. Every customer was overwhelmingly positive with his response, with their methods to keep it clean, and for their honesty. And as far as I know, he has not lost any sort of business at all. He helped create this best management practices guidelines, and really he set the bar, I think, in Minnesota for how garden centers and nursery sellers and other plant sale organizations can really put their customers' minds at ease as long as they are following these best management practices. So, to whittle down this document, the first thing is educate. Educate yourselves, educate your customers, educate your peers. There are tons of extension and agency publications out there. There's ongoing research. There are groups. There are workshops just like this one that will help you stay on top and really make sure that you have as much information, as much of the current information as possible. So don't stop educating yourselves about that. Scouting, that is the biggest thing. That is my number one job. I have to look at all of our different plant communities, all of our different growing ranges, and make sure that I am noticing something that doesn't quite seem right. You want to look for all signs of jumping worms, like if there are castings in your compost or your mulch or even your nursery pots themselves, and check underneath the flats too. If they are growing on bare gravel, like a lot of nursery sites are, lift those up and dig around in that top inch or two to make sure that there are no jumping worms under there. I was talking to Amy in the chat a little bit earlier. Talk to your growers, talk to your contractors, talk to everybody. If they are unwilling to talk to you, have a healthy conversation about jumping worms or other invasive species, or they roll their eyes, or they tell you, don't worry about it, we're on top of it, and don't give you details, fire them. That is ridiculous. There should always be an open dialogue about this. I spoke to our mulch contractor when we discovered jumping worms saying, hey, are you worried about getting compost from us? And they said, we're not, because here's the thing, Erin, here's how we actually deal with compost. We do thorough turnings. We measure the temperature. We are very convinced that every single square inch of our compost has hit 140 degrees for a set amount of time. We are not at all worried about jumping worms. We have never found them in our compost. We will keep looking for them, of course, and if we find them, we'll deal with it at that point. But because we are so thorough in our operations, we are confident that we will never see them. We've been working with them ever since, and we haven't had issues when we go to rebuy our compost from them. It's still very good looking stuff. So talk to your contractors, make sure that you can tell right away if they're trustworthy or not. Only work with the people who are willing to have those conversations with you. And then finally, use iNaturalist or EdMaps to report your new sightings. We're still tracking this and we need help with that. So yeah, just realize that when you put in a report into iNaturalist or EdMaps, at least for the most part, most state agencies are dialed into those apps. And if they have additional questions, they will reach out to you or they will go investigate based on your geographic coordinates or whatever location you put into the app. So it's always good for that to look at that. All right. I know there are more questions in the chat and I promise I'm going to get to them. As far as other helpful guidance goes, check into your extension sources. I worked with the master gardeners to kind of come up with what could we do if people insist on sharing their plants and, we can't really stop them from doing it, but we want them to be safe. We kind of tried to give them visualization. I don't recommend washing in a bucket because you're never going to get everything off of there. But for us, cleaning them off to the bare root, again, when you're washing anything, do it on the same site you're getting the plants from. So that way you are not sharing them along anywhere. You're not moving them off of site, right? And on top of that, any sort of mud like that you're taking off of your garden tools or your shoes or the buckets that you're washing your plants down into, dump that water and mud back onto the site where you got it from. Do not take it with you and then dump it. Just dump it on the original site because you're not going to be increasing your numbers. You're just going to keep it there. And then I saw Angie Gupta is in the audience, or at least was earlier. Angie and her team put together this incredible document on plant sale recommendations to reduce jumping worm spread. Again, I'll put this PDF in the chat for your clicking pleasure, but there are some really nice best management practices for like master gardener or community type plant sales as well. That was kind of what the intent behind that document was. And I still send people to that document all the time because it's very, very good management practices. So now that we're done with the plant sale, I'm just going to take a few more minutes to talk about our forest field trials. We tried to come up with cultural, mechanical, and biological controls that would work in forests because our forest is where we're seeing some of the worst damage that jumping worms are doing. Brittany showed the erosion, so you can see a tree's root flares. It is incredibly exposed. We're seeing that too, and the forest is not regenerating, and there have been other people who are going to talk about that or have already talked about it. So I'm going to keep this brief. What we tried doing was we came up with, and this was funded by both the University of Minnesota's Minnesota Invasive Terrestrial Plant and Pest Center as well as a small grant from the Horticultural Research Institute to continue our monitoring and our trials. And essentially, we came up with a forest at the Minnesota Landscape Arboretum that is infested with jumping worms. We broke it up into 32 plots, and we tried different methods to see if we could reduce jumping worm numbers. Sounds great with intent, right? So since diatomaceous earth has been used on like slugs and other insects, other invertebrates, we thought maybe this could work for jumping worms. And since it's already being used in gardens, you know, maybe off-target impacts are still going to be not great, but maybe it's an option. In the UK, golf courses use sulfur to help with Lumbricus terrestris, the night crawler. So we thought, what the heck, let's throw it in. People use it to lower their pH all the time. We have horribly high pH in Minnesota, so we use this in our landscapes when we need to. Botanigard, as Josef said, he and Miriam had been doing work with Bavaria bassiana, so we thought we would try it as well. Planet Turf tea seed meal, yeah, people know about it. No, it is not labeled. None of these are labeled for earthworms. So again, this is purely research. We work with our Department of Agriculture to make sure that they know that we are conducting research and that we are behaving responsibly. Homeowners can't do this kind of stuff. So, but we wanted to find out if tea seed meal could work in our forests. And we had a few other treatments, which I'll tell you, but essentially our plots were 2.5 meters in diameter, and I'm sorry, the radius was 2.5 meters. And so we broke them up into either 3 or 4 segments that we could evenly distribute whatever treatment into those areas. And there was about a 1 meter buffer division between plots. They were randomized, so that way if we did get to good data, we could publish it. But there's some fun things that happened. All right. 32 plots, 8 treatments with 4 replicates. And it was in a mixed hardwood forest, which was sugar maple dominant. We did a control. We did a little bit of supplemental watering, about the same as was required for Botanigard. So just like 7 or 8 liters. Tea seed meal fertilizer, elemental sulfur, diatomaceous earth, the Botanigard, which is Bavaria bassiana. But then we also did leaf removal, thinking if we took out all of their, the main source of food for them, that maybe they would leave. And then likewise, we decided to do double leaf litter. So cleaning those leaves, making sure there were no jumping worms on the leaves that we were taking out of the plots, and then adding those leaves to another plot and seeing what it did to the populations. We did these treatments in 2021, 2022, and 2024. We had to take 2023 off because we were under severe to extreme drought conditions from 2021 through 2023, and our populations were collapsing. We did pre and post treatment counts that were conducted each year with 30 centimeter diameter PVC rings in 6 locations in each plot. So those population counts took like 2 days for us to do, but we did it at the beginning of the treatments and then at the end of the treatments each year. Soil samples were collected in pre-treatment in early 2021, and then we did it in post-treatment in late 2024 to see how the soils from the very beginning of the trial to the very, very end of the trial differed as far as pH, the percent of organic matter, and bulk density, soil bulk density, because we have seen that jumping worms change bulk density because of the granules. The castings are so loose and do not bind to each other very well. Results were kind of lackluster, and there was no statistical significance because look, from the beginning to the very end of our trial, our control plots lost 3/4 of the jumping worms that were in them. So when you lose your controls like that, your data is not that great. So it's still interesting to see the differences in the plots from the beginning to the end. Our pH changed. These are not the points of pH, so no, we didn't go up 5 whole pH points. It was a percentage. And so, yeah, in some areas we went from like 7.4 pH all the way up to like 7.6, or maybe it was 7.2 and it went up to 7.5, those kind of changes. Elemental sulfur, of course, we saw over a 9% reduction, which makes sense because it is used to acidify your soil to lower your pH. Organic matter went down across the board because, again, we were doing organic matter at the beginning of the growing season when we still had lots of leaves on the ground. And then at post-treatment, that was after earthworms had taken pretty much all of the organic matter away and eaten it. So that was expected as well. Bulk density, this was a bit of a shock and we weren't expecting these results, and we think that it was because there was a couple different methods used. So this was not results that we wanted nor we felt were super reliable. And then again, with the change in jumping worm populations, because the control lost that many worms, and again, it was mostly due to the drought, that's a pretty good indicator that at the very least, we know that drought is a very big killer of jumping worms if you can allow your gardens to survive that kind of conditions. We did see a sizable reduction beyond the controls with leaf removal. So that theory that if you take away the bulk of their food source, they'll either leave the area or won't survive. That could be a practice for home gardeners. Don't mulch your garden beds. Now, mulch helps you with weed control, with water conservation, and to keep your soils a little more cool. Those are all conditions that jumping worms actually like, with the exception of the weeds not germinating. So, I mean, it's an option, but, you know, we would still want to look at it a little more. We did find that Castaway 301 seemed to be promising. So of all of those, you know, those would probably be the two that I would say go ahead and continue researching. Botanigard, we saw some reduction, but not much more than the control. And unfortunately, diatomaceous earth and sulfur, we saw no more control than we would have with we saw no more management than we would have with compared to the controls. So conclusions, yes, drought seemed to work really, really well to reduce our jumping worm numbers. It did not eradicate them. 3 years of drought in a non-irrigated forest did not eradicate jumping worms from that site. It did greatly reduce their numbers, though. The removal of leaf litter and potentially mulch could potentially reduce jumping worm populations due to the lack of consumable organic matter. Mulch will also keep areas cool and wet, which jumping worms prefer. Elemental sulfur and diatomaceous earth did not appear to be effective against jumping worms for outdoor use. So I'm not going to recommend that. Certainly, I would invite people to continue research on that, but I didn't see any sizable difference. Tea seed meal fertilizer, I think, deserves more research for outdoor control based on the greatest population reduction. Botanigard seemed less effective, but I also think based on Josef and Miriam's past work with it, that it still deserves some recognition and some more research. And then complete eradication of jumping worms. If it's even possible, it's going to take a lot of time and a lot more research, so we have to be patient. It's frustrating, but that's where we're at. All right, I will take some questions and I'm going to go ahead and throw this into the chat. All right, it's going to take me a second here.
Thanks, Erin. Let's see. We are looking like Lee has your hand up. You can go ahead and unmute if you'd like to ask a question.
Yeah, I was wondering, Erin, if you looked at how the other insect life and micro wildlife did, you know, the arthropods, copepods, etc., did in these various circumstances, because obviously when we're trying to remove one thing, if we remove all the other things, we're not going to be in good shape. Thoughts?
Yeah, thank you for asking that. It is incredibly important to consider off-target impacts. And that's why we chose the products that we did. We wanted to choose the least toxic options. Certainly, I would think anything like a nasty insecticide like bifenthrin would kill just about everything, including jumping worms. But it's going to kill everything else in the forest too. So we wanted to use things that were already being used as kind of less toxicity options. That's why we went with Bavaria bassiana. Again, as Gary pointed out earlier in the chat, it can affect just about every insect out there. So there's going to be off-target impacts. And that's what we're trying to balance. So no, to answer your question, Lee, we did not specifically hone in on off-target impacts. There are other researchers, including some of the speakers tomorrow, who will be examining off-target impacts a bit more. And so that's of interest for me as well. So thank you for asking that. But that was not our specific protocol for this one. Anyone else want to unmute? Otherwise, I will look through the chat as well. I think Heather was asking, using clean, fresh straw is not any better than using leaf litter as a mulch. And I like that Gary already said that straw, like any organic material, could have cocoons in it. Yes. Jumping worms will still eat just about anything from what I've seen, as long as it has like cellulose in it. We've seen them in pine needles. So even a lower pH environment like that, they don't seem to have an issue with it. So yeah, any sort of organic matter like Gary was saying, yeah. See, I think I see one that says from Nadine, as a manure-based compost business, what are good practices we should put in place to protect our location from materials coming in, like shavings, grass, and our manure? Also, we sell other products like loam and stone, and people come to our location to pick up materials. What things should we put in place to protect our property? The first thing we did is stop collecting leaves. Keep in mind that our location has no power or water. I like that you said that you stopped collecting leaves. So yeah, being really selective over what you allow in, especially with equipment. What we'll also tell people is it's tough because especially if you're an outstate type business and paved roads can be a real problem. Paved roads are actually really, really nice for tires because that does take a lot of mud off of there. It's true that you can't control, this is another conversation I was having in the chat with Amy, you know, you can't control what your contractors are going to be doing. There are best management practices, but unless a law is in place, like Gary said, there's nothing to really stop them from coming in dirty, except you telling them, you just lost my business. I don't want you bringing something dirty into my establishment because you're going to ruin my business. I would equate it to all those organic farmers who are setting up their operations right next door to a conventional farm, and the conventional farms pesticide practices drift over to them and they've just lost their organic certification for 3 years. It's being a good neighbor. It's having these open, honest conversations with your neighbors that's going to probably do the most good for preventing you from finding an infestation popping up because somebody brought something into you and into your site. Fred asked a question, could jumping worms be used for bioremediation in contaminated soils? I don't know that, Fred. There are people who are researching the heavy metal accumulation of jumping worms, but that is outside my field of study.
And you're so quick to find these questions and sort through while you're presenting. That's very impressive. It's hard to do. So kudos to you.
Thank you.
It looks like you were able to grab all the questions that I would have asked you. It looks like there's one more we probably have time for from Jennifer. Do other worms not accumulate heavy metals? And
Yep,
thanks,
Gary got it. Thank you, Gary. Yeah.
I don't even
Gary
need to be
is
here.
quick. I'm impressed with you, Gary. You're on it.
Yeah, thank you, Gary. Yeah, so all worms can accumulate heavy metals if they're present in the soils. If you have done soil testing and you are aware that you know, you have low levels of these kinds of things, then it's okay to feed any kind of worm, especially jumping worms, to your chickens, especially if you're planning on eating the meat or the eggs that come from them. But that's, again, only if you can do soil testing that tests for trace metals.
And then, yes, I want to, Gary, you did answer the question, but I also want to jump onto Caroline's question about that 104-degree temperature threshold. I saw 104 degrees first pop up with Brad Herrick and Marie Johnston studies, where they were studying that temperature threshold with both adults and cocoons. I asked Brad how long, and he said we did it for 24 hours. I don't know if other people have researched 104 degrees with different time already, but I'd be interested to hear that. So if anybody else knows that, please throw it in the chat or unmute. But that's just what I heard was, yeah.
Yeah, Josef Gorres' grad student did this research and found that just reaching that temperature, just reaching it was all it took. That the 3 days thing was an old thing.
Okay, yeah. Thank you.
Just to sort of comment on that too, I've gotten asked that question a lot, because if you're letting it sit out in your driveway, let's say the solarization package, it gets cold at night. So how are you supposed to monitor and maintain those temperatures? And the way that I explain it, after understanding that it really only takes reaching that temperature, and not necessarily that you're not trying to bake it, right? We're not trying to make cakes here. As long as it gets to that temperature at least once, you're okay to have the best chance at killing any of those cocoons that are present. I will say doing it for more than one day, you have a higher likelihood at reaching that temperature, especially if you don't have a soil thermometer. So I'm still letting people know, I think 3 days is a good way, especially if you've got a stretch of that hot, warm sun weather, best way to go about it. But yeah, if you can guarantee that the middle of that soil package has gotten to the correct temperature, 24 hours is good too. Yeah, that being said, it is 3:32, so I think we should go ahead and move on here. So we did have a speaker change. So Dr. Anise Dobson was originally planned to have this time slot, but she has given that to Angela Gupta, who I did see on. So Angela, if you are able to share your screen and get that presentation loaded up, we can go ahead and get to that. Angela Gupta is a University of Minnesota Extension professor of forestry who specializes in terrestrial invasive species and climate-ready woodlands. She's done invasive species early detection education and outreach since about 2008 and led the jumping worms report management participatory science project from 2020 to 2022. Angie earned an MA in organizational management from Spring Arbor University and a BS in forestry from the University of Kentucky. And thank you so much, Angie, for being here. I will go ahead and mute myself and you can take the floor.
Thanks. I imagine you can see my screen and hear me.
Yes, we can.
Perfect. And then I just want to remind you on the backside, if you have my slides, which were in the folder, the front slide, the very first one, has links that can be dropped in while I talk, if you want to put those in the chat.
Yes,
Thank you for having me, everybody. Yep. Thanks for having me. I'm going to talk about participatory science and emotions, solving problems and empowering people. So with that, let's get started. The first, I wanted to define what participatory science is. It is science that engages the public in advancing scientific knowledge by at least 3 things: formulating research questions, collecting data, and interpreting results. And sometimes you can include in there distributing information. And so I've done participatory science that has done all of these things through the years, and all related to invasive jumping worms, rather. So I'm going to talk about this jumping worm project that I ran report management from 2020 to 2022. It started as a 1-year project, and it was really clear there was more to be done. So we continued. So after the first year, I actually brought in and changed the objectives of this project. And so the objectives in years 2 and 3 were, number 1, to provide emotional support to landowners impacted by jumping worms, and 2, gather data from landowners on their jumping worm management activities in order to inform future scientific research and empower others impacted by jumping worms. And so I'll just say the first year, it was only the second part of that, the gather the information about jumping worms. The emotional part was added after year 1 because it was very clear that that was absent and very, very important to my participants. So the outcomes from that first year were a lot of management trials. This is a type of solarization that a participant tried in the picture there. And it turns out gardeners are like immensely creative people. They were very worried about this, and they were doing lots of things. So I essentially said, tell me what you're doing. Please don't break any laws, and then pay attention to the environmental impacts, and we'll learn together. So that's what we did. And we did this in a journal narrative format. So a Google document was what everyone was asked to fill out. We did learn some potential management research outcomes that have been deployed in various ways. And it became very, very clear that my participants needed emotional support and additional resources that I did not have in 2020. So if you recall, that was the first year of the pandemic. The Minnesota Master Naturalist Program turned their volunteers all online, all volunteer training, and they have a capstone course that is required in that program. They were asking for ideas for their capstone projects that could be done remotely and, safely social distancing. And I pitched a lit review to try to understand the emotional impacts of jumping worms. And so God bless them. They went ahead and accepted that challenge, and this would be considered interpreting results in the participatory science framework. So with the help of the Master Naturalist trainees, they did a comprehensive literature review in the scientific literature about the emotional impacts of jumping worms. Yeah, there was no literature at all. So we upscaled that to the emotional impacts of invasive species, still nada. We upskilled that one more time, and we ended up learning about eco-anxiety and climate anxiety, and there was a lot more information there. Once we discovered that field of research, we were able to take parts of it out and apply it to jumping worms and the volunteers that I was working with. So what we learned was that a lot of the volunteers were experiencing feelings of solastalgia. So that is where you mourn a major change in landscape. So the common example of this is if you grew up on a farm and you go back and it's a parking lot, you would experience feelings of solastalgia. But it turns out many people in their home landscapes, when they get jumping worms and they realize that's now a part of their life, they have solastalgia for the time before jumping worms. A lot of our participants were experiencing disenfranchised grief. This is a type of grief for which we do not have cultural norms within our communities. And so really what this meant, they would go up to their local nurseries, and these were commonly female volunteers, and the nursery attendants would give them the crazy lady look. Like they would be very concerned about jumping worms. They would be asking lots of questions. And at that point in 2020, it was pretty common that the nursery industry wasn't terribly well informed. They didn't have any answers, and they were not terribly empathetic to the emotions that were going through the volunteers. I think that has gotten better, but this disenfranchised grief is real. This was also impacting their views of themselves, which is called serious leisure. When you think you're really good at something like gardening, and then something bad happens like jumping worms, and you think, wow, I did that, I brought that in, which I think you need to give yourself grace. But a lot of people were experiencing that, like maybe I'm really a bad gardener, and maybe that means I'm not as good as I was as a person. And these like thoughts were circling in people's heads. And so it was just really important to step back and be like, look, you didn't know, you were trying your best. We're all on a learning curve, and give grace. Then there were some really tangible ways we could make it better for volunteers. So it turns out there are rituals you can do to go through the stages of mourning about the change of your landscape as a result of jumping worms. Hope and agency, we really work to strive to enable that. That is the goal of my presentation today. It is the goal of our work since this time, because there is hope for the future, and we have the agency and the power to make a difference. So figure out how we can deploy that. And then I'm constantly reminding people that there are massive nature-based human health benefits. So whether you're out there, managing your yard for jumping worms, or you're, removing weeds or planting crops, it is very good to have your hands in the soil and be outside. So as a result of all that, we end up creating this emotional support for jumping worms, and I'll talk about that a little bit later, but it is now publicly available on our website, and hopefully the link is being dropped into the chat. We also found in the peer-reviewed literature a survey that we were able to tweak and apply to this project, and so we deployed it to the jumping worm volunteers, and what we learned is that jumping worms cause a lot of sadness, fear, panic, impatience, and anger. Jumping worms increase negative thoughts, and they provoke tears. And truthfully, it was the tears that were really the tip off to me. People would call me, they would cry on the phone, and they would be completely distraught. Another big tip off for me was a lot of people would close their emails to me about jumpings with the salutation, "I'm devastated." And it was just so clear that that wasn't a good space to be in, or a good space for my volunteers. Another really amazing outcome of the project is we learned that just by participating in this project, this participatory science project, they felt and they were contributing to science and the knowledge of this pest, that participants' feelings became more positive, reducing worry, increasing confidence, attention to environmental change, and their jumping worm outreach efforts. So, we didn't come up with all the solutions, but we were a part of the solution, and that itself was very powerful. We learned that this was a big deal back in 2020. The green industry, horticulture, was really worried that when people found jumping worms, they would stop gardening. And that was not at all what we found. So what we discovered was when jumping worm management costs more money and takes time, respondents reported a sharp increase in their efforts. Like they're really double down to figure out what to do about it. And I think this is really important for all the educators and communicators and agency staff on this webinar. As communicators, we must manage the stories and actions we talk about, because those cause anxiety, mourning, anger, hopelessness, and motivation. So to get people motivated, but we want to try to evoke more positive emotions while we do that. Again, there is hope and we have the power to make change. So as a result of all of this, you can find the final report at this Jumping Worm report management website, which again, I think is being dropped into the chat. And you can see the whole report summary for the emotional toll work that we did, the survey results. But I also want to just point out, particularly to folks that work at agencies and organizations, meaningful help is not tricky. So we have at the University of Minnesota Extension a fairly robust Jumping Worm website. And on that website, we have this frequently asked questions section. Within that section, we have added this section. It is normal to feel sad, fearful, panicked, impatient, and anger after learning I have jumping worms. And so then you can click to the report and learn more. You can also jump to this how to manage my emotions, which takes you to this. I've already showed you a quick preview of that before, but it's like a couple of page sort of fact sheet kind of thing about what those feelings are. And so I have a lot of content on our website for all kinds of different invasive species and other things, but this emotional support, this is the thing that people will then take the time to draft a personal email to me and thank me for having. And so I just think it's super interesting that people are feeling sort of so alone and so distraught over this that they take that extra step to tell me how much better they feel when they find this resource. So again, you know, all the agencies and all the people, as you spread the word to your neighbors and friends, like we can support each other as we go through this emotional journey caused by jumping worms. This is a bit of an awkward transition, but I also want to say that we at the University of Minnesota Extension have what we call an invasive species community of practice, and we've done a lot of work to improve common names around many different types of invasive species, but including jumping worms. So we are very intentional about what we call them, and in a process to really come up with this better name, jumping worms, we have these principles that we developed, these guiding principles to try to name these species early before they're well established and kind of part of our local colloquial conversations. It was drafted with the collaboration of our Extension Foreign-Born Affinity Group. Those are some really tough conversations to get us on the right track for developing better and improved common names. We then drafted these guiding principles. The next slide will identify those a little bit. And we continue to do this work. So we've already done jumping worms. That's what I'm working up to. So I want to share with you the words that we're using around jumping worms. But we continue to do other species every year. So these are some of the quick aspects of the guiding principles. We do encourage the use of scientific names when that makes sense. I will tell you, rarely do scientific names used in colloquial terms get all the way to the species. They tend to stop at the genus, so it's only moderately helpful. Many scientific names have either people or places in them, which is also potentially troublesome. And scientific names change. A lot of people think, oh, they never change. They actually change pretty regularly. So they don't often work out great. We want to avoid things like place-based names, derogatory terms, or common names that can be confused with other species. We evaluate all of the common names that we can find currently sort of in colloquial use. And then when we decide we're going to try to work on new names, we do try to improve those names. And so they ideally they highlight the invasive features of each species, if there's any host species, identification species, or symptoms of characteristics of symptoms. Okay, so as I've alluded to, we've actually done like 24 different improved common names, 12 of which are jumping worms. So this is where I've been working up to. So I realize this is going to be up kind of quick. Again, there is a link if you are talking about any of these worms. I strongly recommend you consider using these common names. We've been using jumping worm. That is our recommendation for the Amenthus whole suite. That's a group of species. If you're trying to talk about individual species, which is fairly rare in most conversations, we have tried preemptively to get ahead of those names. It turns out even in their native range, many of these worms didn't have names. And so here we go. And they are oftentimes related to the physical aspect of the worm, again, as a part of our guiding principle. So bottom line, I want you all to be aware that this exists as you talk about these things to friends, neighbors, and coworkers and colleagues. I do a lot of eco-anxiety. So right now, I want everyone to stand up. If I do that, it's funny because you kind of see my belly button, but seriously, everyone stand up. Stretch and wiggle your bodies. We're going to do this practice together once, and then I want you to do it on your own once. Breathe in slowly for a count of 4. 1, 2, 3, 4. Hold for a count of 4. 1, 2, 3, 4. Exhale for a count of 4. 1, 2. Hold for a count of 4. 1, 2, 3, 4. Now do it once on your own. Perfect, everyone's done. So if you want to go ahead and sit back down and get comfortable, I think that's appropriate. I offer this as one of the resiliency practices, or what we call mental flourishing exercises, that we can all do when we're feeling stressed and overwhelmed. Also, I want to point out this picture on the right, this picture of nature and water, is not an accident. There's research out of the Mayo Clinic that combination of picture and of nature and water can be really calming for the soul. And so these are little tiny practices we can work into our day when it's starting to feel stressful and overwhelming. And I think this is really important because as we together as a community, we need to work to build cultural resilience, because eco-anxiety is real. Most Americans have experienced it. So it's important to recognize your feelings, to create rituals to process those feelings, empower hope and agency, and remember the many nature-based human health benefits. So if this is something that's of a concern to you or friends and family or coworkers, I just want to point out that we do have a 2-page fact sheet, and I'm really excited. The University of Minnesota Extension Eco-Anxiety website is up and very robust. So you could just Google and you would find many resources, including the link to this. So I want to just share that since this work started with jumping worms, we have now really worked to inspire hope and agency in all of our extension forestry outreach. And I do a lot of participatory science. And so this is sort of this year's suite of species and things we're working on. And you can see right at the top of this jumping worm soil erosion project. There was a 3-year research project that we have state funding to do. My next slide will talk a little bit more about that. So we're actively recruiting volunteers if you're in Minnesota. I'll say more about that in the next slide. And then right after that, I'm going to talk a little bit about this, the Tree Steward Journal and the Tree Tracker. And I'm going to close with what I hope is an upbeat story. And so I will move along. But these are many of the ways in which we engage participants and community members to further science as we go. So I alluded to this, jumping worms and soil erosion in Southeast Minnesota forests. This is try to understand. Erin talked about how we're seeing a lot of negative impacts in a forest and we don't have a lot of great management options. So we're really trying to understand what erosion is happening. And so I am delighted to have a new colleague, Hannah Kim, who is the leader of this work. And so she's currently recruiting volunteers. So again, if you're in Southeast Minnesota and interested, please reach out to Hannah. And you can go to that link and find lots more information. So after I learned about eco-anxiety and really started applying it to our work, we in Extension Forestry created the Climate Ready Woodlands Project. And I'm not going to go into a lot of detail about it, but ultimately, after 2 iterations, we ended up with 11 different ecoregions. Each ecoregion has 4 different lists, and those lists are climate ready native trees, tree species to consider as refugia. So those are tree species that are native but unlikely to do well in our future climate. And it turns out that we didn't have the first time around, and not having that list of maybe sometimes beloved native trees, that caused a lot of anxiety. So we had to go back and retweak and add the refugia list. Then we have native understory plants that are likely to do well in our future climate. And finally, we included migration potential species. So these are trees or plants that are likely to do well in our future climate, but are not necessarily native. They are not native in the ecosystem in which that they may do well in the future. And so all of the trees and plants on all 4 lists are known. We have documentation that they are beneficial for little critters in Minnesota. So think like pollinators. But it turned out that this migration potential species caused a fair bit of anxiety, right? So can it survive? Can these species, new to region species, survive? And will they become invasive? Were kind of the 2 big things. So we're running a bunch of participatory science projects to try to understand that. And one of the core principles of this work, I'm going to talk about and have an example of. The example is from Rochester, and I've starred that. It's in the southeast corner of the state of Minnesota. And the core principle is really based on this idea of rewilding. So again, I'm going to show you this example of the Plummer House, which is a park in the city of Rochester. And it was originally a very large home and property owned by one of the founding doctors of the Mayo Clinic. And so this idea of rewilding is interesting. So rewilding, this is a long, complicated definition. I'm going to read the bolded parts. Rewilding is increasing biodiversity and restoring natural processes. Rewilding aspires to reduce human influence on the ecosystem, and rewilding is open to novel and emerging ecosystems, which encompass new species and new interactions. The shorthand is rewilding is creating resilient, self-sustaining ecosystems for the future. And why does this matter and how is it related to jumping worms? This example that I'm going to show you has jumping worms on the site. So I'm going to sort of talk you through it, and then hopefully you'll feel better about the future. It's not all doom and gloom, although it certainly can feel that way at times. So this parcel has two main slopes. There's a north-facing slope, which has jumping worms, and there's a south-facing slope that has non-native earthworms. Like Maine, there are no native earthworms in the state of Minnesota. But on the north slope, this is some of the trees on the north slope. We have eastern hemlock, which is up here in the far left corner. That is considered very rare to threatened in the state of Minnesota. It is considered native, but this is a relatively young population. We have way too many Norway maple, which is considered invasive, and we're slowly trying to get rid of it, but they're kind of older, and so it's a little bit of a heavy task. Then we have quite a number of eastern white pine, which are not regenerating an understory because of deer browse, but they are an important overstory characteristic. Interestingly, we have sycamore, which is not considered native to Minnesota, but it is on the climate ready list. And so it's likely to do well in our future climate and it's considered migration potential. And then we have eastern black walnut, which is a lovely native species here in southeast Minnesota. It is considered migration potential in northern Minnesota on Lake Superior in the Arrowhead. And it is likely to do very well in our future climate. In addition, we have things like Ohio buckeye, which most sources would say is not native to Minnesota. Some say it is. But it is reproducing and successfully growing on a jumping worm infested slope, and it is not getting eaten by deer. So that feels very promising. We also have we have cherries, a number of cherries. We have alternate leaf dogwood, and then we have elderberries. And so this is it's a very weird plant community. It's not considered a native plant community, which we have good documentations on. This is why I would consider it novel. And there are jumping worms on this site. On the south slope, it's very different. South slopes are much drier in Minnesota. They're much warmer. And so we have some eastern red cedar, which I think is a residual. We have some white pine. We have more black walnut. We have some honey locust and those wicked big thorns, and that is reproducing through sprouts, which is interesting. We have a remarkable volume of hackberry, which is super weird. And then we have black maple, which is interesting. We have a lot of little things in that understory, many molds, as I've removed lots and lots of invasive species, columbine, we have jack in the pulpit, we have woodland sedge, false Solomon seal, and then some interesting sort of prairie viburnums or prairie trilliums. And why do I think this is maybe even considered a successful novel ecosystem? This is why. This picture was from 1940. You can see the aerial photo. You can see the south slope was what we would call a goat prairie. It wasn't a forest at all. So in the last not quite 100 years, it has converted to a forest. And this, I believe, to be the old original driveway, which is why I think there's some interesting species like the sycamore and the Norway maple. This is what that site looks like today. Both are clearly forested. So when left to sort of nothing and benign neglect over time, it has transitioned to forest. But here is where I think the rubber hits the road in the tropic triangle. So we have a lot of critters living in this very small inner city urban space. We absolutely have a very robust, troublesomely robust deer population. We have fox eating rabbits. We have a fisher. So the fisher, this was the first report of a fisher this far south and west in the state of Minnesota when it was made. And fishers are expanding their range southward, which is super interesting. So they're not truly considered native in this area, but they are on the move. Of course, we have raccoons. This bald eagle, we had a nesting pair of bald eagles move into a white pine on the site this year, and we believe they successfully hatched their eaglets. So this would be the top of the tropic triangle. There are many other raptors on site, including this hawk. We have lots of squirrels. We have breeding turkeys, rabbits, lots of birds. We have possums, which are on the move from the north. We attribute that to climate change, but they have been moving further and further north in Minnesota. Lots of birds. And then a suite of many smaller things, including native eastern bumblebee, non-native worms, non-native but beloved honeybees, and then the red emerald butterfly. And so all this to say that when you work to steward an ecosystem, I think there is hope, even if it is, it has a lot going on. And so with that, I will take questions, and I like to field questions while I have an inspirational slide up. So this is the south slope of the Plummer House when my family started working there in 2020, and my boys weren't quite teenagers, just got sent home from school. This is an understory full of invasives. So think honeysuckle, buckthorn, Japanese tree lilac, garlic mustard, dame's rocket, motherwort, non-native worms. A year later, these are all the native eastern bluebells emerging in the spring. You can see 4 years later, all of that material has been knocked down. None of the invasive plants were ever taken off the site for logistics reasons. And then the fall last year, all of the beautiful white snakeroot that is native, and deer won't eat it. So with that, I will stop talking and take questions. And I am not as good at Erin and reading the chat. So if people want to stop and feed me questions, that would be great.
Thanks so much, Angela. That was amazing. I think I'm going to take that box breathing mechanism into my day-to-day, especially working as an entomologist with all the forest pests we do. It's definitely helpful to run through that. So thank you for that.
Yeah.
Yeah, so I was looking through some of the comments, but it looks like there's a lot of commentary. So I would invite, if anybody has any questions, to go ahead and unmute themselves and ask, and then I'll continue filtering through the chat here.
And Brittany, just looking at the time, it's 3:58. You might want to toss that SAF in the chat, and then I'm going to share my screen, Angela, and just put up a QR for folks that are here for credits.
Yes, I'm trying to read the chat, but I think raising hands and unmuting would be delightful.
Looks like Lee has her hand up, or his, not sure. Has her hand up, or his, not sure.
I'm a she, and my question this time around, Angela, is I was just super curious when you said about hackberry, why it was super weird, because I love, I work in the ecotonal area, let's say, and around invasives and natives and kind of what you're talking about. So I'm curious why that was the one you were like, this is super weird.
It's the concentration on that slope. So it is one of the species. Yeah, so maybe mute to get rid of the echo. Perfect. So that slope is the dry southern slope. And so it's not that we wouldn't necessarily see a few hackberries, but it's a little uncommon on those dry south slopes. And there's a lot of it. I would argue, I haven't actually done the inventory to count, but it is probably the dominant tree in that stand, which we don't see. It's not typically a dominant tree. And it doesn't appear to be getting eaten by deer, which I think is its reproductive success. It is a wonderful species. It is currently my favorite. You know, you can eat the fruit. I enjoy it. I offered at the holiday party. All of my coworkers got hackberry milk as a sweet treat. And it is great for wildlife. It is very robust, but it is odd in that concentration. And so one of the things when I realized that I was working pretty regularly in a novel ecosystem is the fact that it was a weird conglomeration of species. Not that any of them individually were troublesome or concerning necessarily, but they were a very weird combination for the state of Minnesota. And I think that's because it was just a neglected site for a long time, and it was already converting to these climate resilient species without us really paying attention. And then we did this whole separate project. Oh my gosh, here are the climate resilient species. And wow, when you really start paying attention, that is kind of what you're seeing in these neglected areas. So this was an extreme example, but I think it's the only one that we have thus far of a truly novel ecosystem. So great question. It is a lovely tree though, hackberry.
So Justin Richardson is a soil biogeochemist who studies the movement of elements in soils. He has studied nutrient and toxic element cycling across the northeastern United States, particularly northern New England, for the past 14 years. He was an assistant professor at the University of Massachusetts Amherst, but is now an assistant professor at the University of Virginia. And Justin, without further ado, I will go ahead and hand you the metaphorical mic here, and you can go ahead and get started. Thank you.
Awesome. Well, thank you everyone for coming out and for a talk on jumping worms and soil nutrients. I wanted to differentiate myself from all the other talks because a lot of the other talks are probably more pertinent about control and their spread and where they move. So I wanted to focus on one of my expertise, which is soil nutrients and how earthworms, specifically jumping worms, will affect them. And I'm super happy that you guys all logged in and you tuned in. I'm very pleased to have friends here like Ennis Dobson and some of the people I've looked up to, and fellow biogeochemists like Ivan Fernandez in the audience, and then some of my current students are in the audience. So it's a lot of fun. All right, so let's see about hopping in. Okay, great. So as mentioned, I am a professor at the University of Virginia, assistant professor, and I focus on soil biogeochemistry. I look at nutrients and toxic metals. So if you look at my studies, you'll see emphasis on both toxic metals and nutrients. And as a biogeochemist, I'm not a biologist or earthworm scientist. I look at how interactions with rocks and minerals form soils, and soils interact with plants and natural waters, and then how that impacts humans and animals and earthworms. So I take this really integrated approach across the different disciplines to understand how elements move among soils, rocks, minerals, plants, waters, and animals. So I'm not a biologist, I'll say that again, but I have been worm hunting for a while. I just remember digging in soil pits in northern New Hampshire in the White Mountains and finding earthworms pretty abundant, which is something that wasn't really in the literature at that time very clear. And I've continued earthworm hunting with even my current students like Sophia Hardesti, looking at these invasive earthworms and exotic earthworms in Virginia. So this talk has 4 parts to it. First off, I wanted to really hammer home exotic and invasive earthworms yet again. I know that some of my predecessors gave some good insight, but I wanted to take a broader step back of the biology of it, look at jumping worms and how they differ from the other earthworms, specifically the European earthworms, and then get into their impacts on soils. And lastly, what you came here for probably is their impacts on nutrients. So first we'll start with exotic and invasive earthworms. So when do we see earthworms, right? We mainly interact with earthworms after a good spring rain. They all pop out from their burrows and their little hiding cubby holes to stretch their segments and go for a little walk now that it's moist and warm enough for them to hit the streets. We also see earthworms and use earthworms as fishing baits. So if you're ever going out fishing for whatever species, earthworms are a very common popular bait. They wriggle and the perfect texture for fish to hunt. And then lastly, maybe you're into vermicomposting. So maybe you have some Acenia fetida, red wrigglers in your compost to help decompose organics. But it's actually worth taking a step back and look at the biology of earthworms. What are earthworms? So if we look at the tree for classifying animals taxonomically, on our far left is our most simplistic animals like porifera, like sponges, and then we get to our cnidarians. We see platyhelminthes, so that's like our freshwater flukes. We can see nematodes, our false worms, and then we see shelled organisms like mollusks, like clams. We see annelids are actually pretty far to the right, meaning that they are pretty advanced in their structures. So here, chordata, shown by this frog, is where us as animals live, humans with vertebrae. And you can see that from the ancestral evolution that they have bilateral tissue symmetry, a true body cavity. They're protostomes, not deuterostomes. They are coelomates, meaning a true body cavity, and that they're actually pretty evolutionarily advanced. So they're not nematodes. They're not flukes and platyhelminthes. They are true worms. Great. So there is our annelids there, denoted by the earthworm. Earthworms actually have a lot of structures, as I was getting at. They have a mouth. They have several heart chambers that move blood through their circulatory system. So if you cut a worm in half, it will die because there's one part with the heart, and the rest of it is the digestive tissues. You see earthworms have male and female reproductive organs. That's why they can have parthenogenesis and lay cocoons. They have a gizzard for grinding up materials. Here's their saddle/clitellum, which is used for identifying them and counting the segments. They have blood vessels. They have a true digestive system. They have a actual a nervous system, they have intestines. They're actually pretty advanced structures in earthworms. Okay, earthworms are typically detritivores or omnivores, meaning that they could either eat soil organic matter or the biota that's feeding on soil organic matter. There's over 3,000 species worldwide. There's some fun facts like earthworms can digest and ingest 2 to 30 times their own body weight in soil every day. And yes, cutting a worm in half kills it because half of the worm has the heart's reproductive structures and the other half does not. When we like to think about earthworms, we like to think about their feeding groups, aka their ecophysiological group. I think this was mentioned before, but we have our epigeic earthworms, which are small litter feeders. We have our endogeic, which are mineral soil feeders. We can have our anisic earthworms, which makes permanent burrows, so that's like our Lumbricus terrestris. And jumping worms really fall into this epiendogeic category, which is that they can both feed and live in litter, but they can also feed on the mineral soil. So that's where our jumping worms live, as both a mixed epigeic and endogeic. Great. So just looking at their consumption, so on the left picture, we can see an anisic earthworm, probably Lumbricus terrestris, consuming leaf litter. They almost always grab leaves by their petiole, which shows that they're really eating leaf litter. Our epigeics live in the litter layer of the forest, and they eat the litter. And then you have your endogeic earthworms, which mix and live in the mineral soil horizons. Earthworms in New England were largely wiped out by the Laurentide ice sheet. I know that there's a lot of talk about some of the native earthworms. There are some pockets of native earthworms where they've been reintroduced or brought up north, but largely the forests of northern Vermont, New Hampshire, and Maine largely have exotic earthworms that have been brought up by humans. So most of the native earthworms have been pushed down because of the glaciation down to New Jersey, Pennsylvania, Virginia, Kentucky, Missouri. So we don't have a lot of native earthworms in New England. If you look at even like their spreading rate, their natural spreading rate of either crawling or having a bird pick it up and drop it off later, it's not very far. So it's really through human actions that earthworms have been moved around. Cool. So that's our understanding of like our earthworms, our exotic invasive earthworms. We're going to hop into what are they and how do they differ in our second section. So most of the earthworms that we'll see in forests are a lot of European earthworms. They were brought here by the European settlers. They actually brought them in soil that helped act as ballasts from their ships, and they also brought them for their crops. So those two ways brought a lot of earthworms, and we've since moved them because whenever we trek soil or their cocoons are stuck to equipment, they just get moved around. So the European earthworms come in two main varieties, our pigmented and unpigmented earthworms. So our pigmented earthworms are commonly found that are Lumbricus terrestris, aka our Canadian nightcrawler. By the way, I hate common names just because they get thrown about in so many different ways. Here's our Lumbricus rubellus, aka the redworm. It's also Lumbricus, so it's related closely to terrestris. And then we could potentially have, say, Dendrobina hortensis, which is the European nightcrawler. So a larger earthworm is striped and banded. So these are some of the common pigmented earthworms we see in New England. Other earthworms that we see are non-pigmented. So these are our endogeic earthworms that do not come to the surface. So you usually only find them when you dig. These are typically like Aprectidea, so Aprectidea rosea, our pink worm. You can also have our Aprectidea caliginosa, a type of gray worm. And then we can have our Octalasian cyanium, which is slightly blue and has a yellow tail. And these are all endogeic earthworms, and they're not pigmented, so they do not come up to the surface very often. So whenever you're digging, you're likely to see these, especially once it rains. They're very common to come up. But the earthworms that we're really interested in are jumping worms. So unfortunately, to be more scientific, I'm going to really refer to them as our metaphyre and amynthus earthworms throughout the rest of the presentation, because those are not the common names. They're a little bit more taxonomically correct, even though that they're actually pretty broad, and there's still a lot of debate on species, where they begin and end. Okay, so with the amynthus and metaphyre earthworms, they are segmented earthworms, of course, they're true earthworms, and they undulate like a snake. So they have a high hydrostatic body pressure, which allows them to wiggle like a snake rather than our segmental undulations. Again, they're epiendogeic earthworms, meaning they eat soil and litter layers. They hatch in the spring and die in the winter. It's pretty much programmed that they die. They're parthenogenic, meaning that they can self-fertilize and lay eggs, but they can mate. And they are prohibited in several states to own and propagate, but that's not all states, and propagation has been pretty common in southern states, in the United States. Okay, the main jumping worms that we find in the Northeast and New England are our Amynthus tokyonosis, which is our smaller of the Amynthus metaphire. Then we have our Amynthus agrestis, which can range from smaller to chonkier. And then we have our largest of the jumping worms, which are metaphire Hilgendorfi, which are very large, 7 inches. Once relaxed, I've had one stretch out almost to a foot long. They're pretty gross and long. So they range in size and they range in species. And to correctly identify them, you will need a couple closer inspections for some of their sexual organs and structures to really make the identification. So largely, I just kind of hang out at the general level of just Amynthus and metaphire. Okay, so why are metaphire earthworms different than European earthworms? Dr. Josef Gorres, he gave a video recording yesterday. We've been hunting these earthworms since 2012, and he's been doing it probably 5 years before that. They're just abundant and very different in certain respects. All right, so the metaphire and Amynthus earthworms are larger than many of the European earthworms, and they're typically more abundant. So from some of my work, my PhD, where I did 45 forest stands across Vermont, New Hampshire, excavating and doing hand sorting for earthworms, because the spicy mustard is not my favorite. I like to go down and hunt them myself. We were looking at sites that are pretty rural, so they're not urbanized. They're more like near lakes where there's a little bit of landscaping and parks, and then some of the small rural villages. So this matches what you see in a lot of rural Maine. But we're finding that the earthworm abundance was pretty high for several of these species. So some of the earthworms like Amynthus agrestis, which included a couple Hilgendorfi and tokyonosis. Then we have our Apractidea rosea, again, our pink worm. We have a Apractidea tuberculata, and then we have our Dendrobina octaedra, which is a small litter layer earthworm. We are having like 50, 60, up to 70 individuals per meter squared. So that's a pretty high density compared to, say, our Lumbricus terrestris, which is less than 10 individuals per meter squared. But when you weigh the earthworms by their dry weight, they're actually really large, especially the Hilgendorfi. So their individual biomass, when combined with their earthworm density, creates actually a pretty large biomass of hungry earthworms. So here we can see the Amynthus agrestis having higher dry weight per meter squared. So due to their high abundance and large body size and their explosive populations, they're pretty different ecologically than our Lumbricus and other European earthworms. So I've done a lot of work also in urban areas. I mean, we live in typically more urbanized areas. And one of the cities I was working in, looking at kind of pollution sourcing, was Poughkeepsie, New York. Poughkeepsie, New York is pretty much a wannabe part of New England. It's pretty close to Connecticut, Massachusetts. And in 2018, I sampled 28 urban forest sites to look at Amynthus and metaphire, and we found at 22 of the 28 forests that were selected across the city, that we found the earthworms up 22 of them. So they're pretty abundant. We were finding them at the edges of golf courses. They were in wooded landscaping and multifamily housing. We're finding them in business areas, and then also at the edges of woodlots and agricultural fields. We were finding them in very diverse ecosystems. Well, at the 22 sites, we were finding that the earthworms had a pretty high density, but a big range. Here we're comparing Amynthus agrestis and the larger metaphire Hilgendorfi, and we're finding a pretty decent population across both the agricultural, commercial, some of the parks and reserves, and residential areas. So the land use, even though one would suspect that there are transport and movement by landscaping and ornamentals would be driving it, but clearly we found them across all these different land uses in urban areas and not urban areas. Okay, so section 3, looking at their impacts on soils. The forests of New England have largely developed without earthworms, again, due to the glaciation and the mile of ice that covered New England. They were largely expelled. Okay, so the soils of New England range from our young inceptisols, so these are all glacial soils with pretty decent rock content. Then you can have more organic, rich, and acid soils. So these are our spotosols, which have this nice E horizon, which are pretty acidic. And we are finding earthworms across all of these different types of soils. Here is a picture of me holding a stretched out Lumbricus terrestris, and this is about a 10 centimeter interval, so that's a 20 centimeter long earthworm, so shy of a foot. So we're finding larger earthworms, and both Amynthus and European earthworms, even in acidic soils that you find in New Hampshire and Vermont and Maine at like pH 3.8. So the acidity was not necessarily a barrier to their invasion. And of course, by eating the litter layer, which we'll get to in a second, they actually alter the soil and increase the soil pH and make it more susceptible for further earthworm invasion. And this has been seen from Maine to Wisconsin. Wisconsin has even seen this in the 1960s, a couple of papers published on that. So earthworms, they disturb how the natural soils form and their processes in New England. So by consuming the organic horizons, they create vertical burrows, they decrease the thickness of the forest floor / organic horizon and make it very thin. So when we have earthworms invading the soil, it just behaves and looks differently. And then when we have our earthworms, specifically our jumping worms, our Amynthus emetophyre earthworms, they actually completely lose their organic horizons and make a deeper A horizon. So this change in soil horizons really alters how carbon and nutrients can be stored in soils, which we'll see in a little bit. So by eating the organic horizon, aka the forest floor, this alters how forests behave because the forest floor is important for a physical barrier for maintaining moisture. It has a very low fluffy density for germination and roots. And then it also has typically a higher nutrient concentration and content than, say, the A horizon or E horizon. So that's really upsetting to native plants and their germinations. So in a typical area, when you don't have earthworms, you get nice, thick understory plants like seedlings. So this is in Vermont, maple seedlings, and blue bead lily. This is over in the White Mountains of New Hampshire. We get lots of trillium and trout lilies in the understory when you have that nice thick forest floor and organic horizon. But by having the invasion of these emetophyre and Amynthus earthworms, they consume the organic horizon. You can actually have grasses sprouting, and you lose the forest litter layer. All right, so this brings us to the nutrient impacts. It's really important to consider that earthworms are in agricultural areas and they potentially have positive impacts on agricultural context. So I want to split apart agriculture from forests, which I'll come to in a second, but I've been looking at earthworms slightly in the agricultural context more recently. This is a picture of Cedar Circle Farm in Vermont along the Connecticut River. And, earthworms in this agricultural context is actually very important, both for how nutrients are affected, but also water and density, and also their ability to crop and not have any damage to their rooting systems. So there's a lot of unknowns. If you came here for answers with agricultural nutrient cycling, I wish I had answers for you. Unfortunately, my presentation will probably bring more questions up. But there are some trends and generalizations from their European counterparts that we can at least start to predict how are Amynthus emetophyre earthworms going to impact agricultural soils and nutrient cycling. Okay, so in agricultural fields, earthworms can do some good and some bad, right? There's definitely positives and negatives. So by having crops and their leftovers, and then also the mixing of the mineral soil, earthworms can do various things. First off, earthworms are eating the organic matter. By eating the organic matter, they're going to respire and release CO2 from soils. In their gut microbiome, they actually harbor nitrogen and denitrifiers. So you actually get releases of nitrous oxide coming out of soils. Earthworms have been shown in many pot studies that they can increase stable carbon. So those are the conversion from more initial decomposed to more stable carbon. They also have calciferous glands, so they can actually increase the pH of soils by eating the organic acids and organic matter that create the organic acids. And by destroying and turning over the organic matter, they can actually increase the availability of inorganic nitrogen. They can release phosphorus, potassium, and calcium for plant and crop uptake. So those are some net benefits. But by decomposing organic matter, they can actually increase dissolved organic carbon in the soil leachate. And by disturbing the sequestration and cation exchange capacity of soils, they could actually release dissolved nitrogen and phosphorus into waterways and out of the soil. So that's a net negative for cropping systems. So previous studies have shown that earthworms decrease bulk density. So you can actually have more aeration and porosity for water infiltration, which is good in agricultural context. Anistic earthworms create these vertical burrows that can both help prevent clogging of and waterlogging of surface soils and improving soil drainage. The study actually measured 240% increase in water movement, so that's a net good for agricultural soils. But another study found that Lumbricus terrestris can actually make burrows that connect to tile drains, which means rapid loss of any fertilizers that are applied to the surface. So if you have like biosolids or animal manure, then potentially that will rapidly cycle and cause eutrophication. So, you know, there's always good and bad with earthworms. When we're thinking about the decomposition of plant residues, earthworms rapidly decompose plant residues, right? They got to eat something. So that can be the stabilization of carbon and nitrogen soils, or can actually be the emissions of carbon dioxide or nitrous oxide as a gaseous form. Some of the Aprechtadias, they've actually been shown to increase organic matter, again, through the mixing and stabilization of plant residue carbon. But of course, the breakdown of plant material can mobilize nitrogen and inorganic nutrients, which can either be rapidly taken up by plants or released and lost. And then there's also this aspect that earthworms ingest seeds. So earthworms can actually increase the germination rates for certain seeds, but also can decrease the germination of other seeds, depending if their gizzard can actually scarify and destroy the seed coat enough for promoting germination, or if it actually then causes the seed to be decomposed. So there's a lot of questions on that. And then also earthworms can potentially affect the fungal communities. So if you have symbiotic fungi, that might be bad for plant roots, your mycorrhizal fungi, because the earthworms could actually eat it as a, you know, in their omnivorous actions. But also earthworms can help potentially allow for mixing. So, you know, it can go both ways. I wish I had answers on this. But of course, earthworms in the forest context is generally bad. So earthworms, again, are eating the litter layer and they're eating the mineral soil. They're, of course, releasing CO2 and nitrous oxide. They can, of course, increase the availability of nutrients. They can also cause the loss of nutrients from the forest soils. So going back to some of the work that Dr. Anise Dobson, who's in the audience, shout out, we wrote this paper together and on some of her PhD work. And essentially that when you have earthworms that are, an earthworm-free understory, that there's a tight nutrient cycling where the litter layer holds nutrients, it slowly decomposes, and that allows for slower uptake and tight nutrient cycling. But once you have earthworms eating the organic horizon and mixing, it can create a stressful rooting zone. You get rapid buildup of exchangeable nutrients, which can either leach out or be utilized by plants that can really take up lots of nutrients very fast, which are typically your more invasive plants. So there's a potential feedback for invaded earthworm plots to rapidly turn over nutrients, which promotes certain plants that can take it up, while our native plants like trout lilies and Trillium may not be adept for that nutrient burst, and also the loss of forest floor. Great. So it's really tough to study earthworms, and especially the metaphyre and amynthis earthworms, because they are spatially heterogeneous. So the complex spatial assemblages of earthworms makes it really tough to study. Essentially, they're dynamic through time. So you can have earthworms in one location. You know, to be able to get this high resolution data is, you know, pretty tough. But within a couple of years, they can actually change their distribution where they live. So being able to ascribe specific differences and changes to the forest at a bulk scale can be done. but finding the individual changes is difficult. So we actually oftentimes have pretty high error bars in field studies trying to ascribe what are changes from our Amynthus emetophyre invasive earthworms. So oftentimes we have to rely on pot studies where you just put a bunch of earthworms in a pot or a bucket and see what changes. But of course, those are artificial conditions and doesn't necessarily replicate what you have in terms of hydrology and temperature and rooting structure in forests. So any study that extrapolates from pot studies to field scale, should really take those results with a grain of salt. All right, so some final thoughts. Who are some of the winners with invasive earthworms, specifically Emetophyre and Amynthus? Well, the increase in soil pH and the rapid decomposition of plant residues can really bolster certain plants to take up nutrients. This is a picture actually from my backyard in Massachusetts, where we had these Amynthus earthworms hanging out in a yard. Obviously, if I have clover and American violets and such, like in crabgrass, it was not the best yard, but they were present. So there could be potential winners from the rapid cycling of nutrients. Some of the potential losers might be, or gain, might be ornamental plants and horticultural plants. If they're not well studied, we don't have a lot of information on how ornamental and horticultural plants benefit or are impacted. You can have root effects because of course, the earthworms eating their symbionts and disturbing their fungal networks. You can have impacts from nutrient losses from rapid cycling of nutrients that leach out the bottom. If you have mulching that's there to protect the surface evaporation, you can potentially have increased drought stress because you're having increased water loss from evaporation. But again, you can have potential benefits from greater nutrient availability for certain plants, higher pH, which of course reduces the amount of nutrients being lost. You can have greater water infiltration and less overland flow, and the reduced bulk density can promote greater root growth. So it's a mixed bag. I wish I could say that there's going to be clear winners and losers. All right, so with that, I guess we'll open up the discussion. Again, I'm a biogeochemist, a soil biogeochemist. You can go to my website if you want to download any of my papers, but I'm not a taxonomist, and I'm not on the control side of things. So I'll do my best to answer and field whatever questions you have. With that, thank you. Sorry, yeah, I talked really fast. I want to make sure that there's time to have questions.
Thanks so much, Justin. Well, the good thing is it is being recorded, so you can always rewatch, pause, intake the information, look at the photos a little bit more. You had some great graphs in there, so thank you so much for your presentation. There's been, again, like yesterday, some really good conversation in the chat. Gary has, again, really been on top of all of the comments and questions in there. So if you do have any questions, feel free to now raise your hand. I see that we have Nancy here. So Nancy, I will go ahead and allow you to unmute so you can ask Justin your question.
Yeah, thanks. Thanks so much, Justin. That was great. I am curious your thoughts on in the Northeast, which of the impacts of these jumping worms are the worst for native plant regeneration? I heard you say increased pH and consumption of the leaf litter O horizon. Are those kind of in combination what is harmful for native plant germination, or is it more than that? Is it one thing more than the other?
Yeah, great question. I'll do my best, Nancy. So the consumption of the forest floor, which can be anywhere from 2 to 8 inches thick, is really important in northern forests for the germination and rhizomes and the tubers of many of the native understory plants. So by losing that layer, I'm not sure how successful a lot of those understory plants are going to be living in the mineral soil. Not only that, it changes the hydrology, it changes the nutrient cycling, the temperature. So I know that there's probably going to be some winners in that sense. And if there's any plants that need that strong acidity from the organic acids that are generated through just the microbial decomposition of the forest floor, then they're going to be negatively impacted as well. So that will promote, I mean, in combination with climate change, I think a lot of the softwoods are going to be outcompeted by the plants that do well with higher pH and less organic horizons.
Okay, and so by the organic horizons, you're talking about both the leaf litter layer. Are you also talking about the top mineral soil horizon, or?
Yeah, no, yeah. So yeah, unfortunately, I'm like interweaving different terminologies. So the forest floor, in my regard, is the leaf litter, so the OI horizon, the partially decomposed OE horizon, and also the humified OA horizon. So some people lump the OA horizon as a mineral soil horizon. It technically isn't, but they will all be impacted.
Cool, thank you.
Yeah, thanks. Great question.
Thanks, Nancy. Jennifer, I see that you also have your hand up, so I will also allow you to unmute. So you can go ahead and do that and ask Justin your question. Unless it was a mistake, that's always possible too. But Jennifer, I do have you able to unmute. So if you are talking, you do have to manually unmute yourself using the microphone button at the top.
While she maybe works on figuring that out, we have Jody in the chat who said, I'm finding the jumping worms in our green stormwater infrastructure systems, which are rain gardens, and I'm wondering if anyone is looking at how they may be changing the efficacy for stormwater treatment within these systems.
Great question. So I have found Metaphyra hilgendorfi along waterways in the Connecticut River. They seem to do pretty well in wet areas. And of course, they're just mixing and eating all the organic horizon that they, or organic material that they can in those wet soils, which is pretty, pretty high. That is a great question. I'm going to probably go on. It depends on what's in the stormwater system. So if it's something that's going to not be decomposed, then their impacts might not be so substantial. It might be negligible. Depends on their population size. If you also are using things like biochar and some of the other materials that are a little bit more resistant to earthworm decomposition, then you might not lose that kind of capacity for storage. And then also, I would really think about the plants that are present. So I didn't have a point to mention here because I don't have any data, but it looks like grasses that are in really high density and really thick make it really tough for them to move around. So if you have like more cattails, typha, or sedges that are kind of really strong bunchy plants, I don't think they're going to impact the nutrient sequestration uptake. But that is another question I don't think we have answers for yet. So thanks, Jody. Great question.
Do you want to try again on unmuting, Jennifer?
I think she wrote it in the chat that I did understand that you have found these worms since 2012. Yep. And whoever you were working with, at least for 5 years prior. Yep, that's all Josef Gorres, who gave a talk yesterday. So these earthworms have been around 20 plus more years. So yeah, most likely, yes. I think I'm trying to remember that one of the first observations was when they brought cherry trees to Washington, D.C., that they were noted in the early 1900s. That was one of the first confirmations. And Gary just posted that some reports in Maine in 1950s. And then the extension for Jennifer's question, has evolution happened? Unfortunately, it's too short for some of the microevolutions, although because of rapid population turnover, it is possible that they might evolve quicker than other species. Oh, looks like Bonnie might have a question.
Yes, Bonnie, you should be able to go ahead and unmute. So it'll be up at the top of the web browser. It should have a microphone with a little cross on it. You click it once, it should allow you to unmute yourself. Nothing. Okay. If that's okay. Are you able to see the chat? Maybe you can type your question if that's easier for you, Bonnie. Yeah. Okay. Sorry about that. Technical difficulties for everyone. It's the means of working in a virtual environment.
We can't unmute people. And I guess maybe that's a privacy feature. While we wait, Liberty Library asked, why do invasives tend to be able to take advantage of earthworm affected soil?
Yeah, I don't know if Dr. Joseph Gorres mentioned this yesterday, but in their native range, the metaphyre and amynthus earthworms live in disturbed areas, so they are really adept to areas where they have landslides and in Japan and Korea and massive erosional events. So those are perfect habitats that they've evolved for. So when they see these disturbed soils or they have similar types of texture materials here in New England, then, or there in New England, they're able to rapidly take over. Same thing with, say, Japanese knotweed and garlic mustard. Those are all plants that have evolved for, like, flooding events and landslides. So that's what makes them adapt. So typically, invasive plants are ones that are very plastic, ones that are able to adapt to new environments quickly. So Bonnie had a question about salt water. Now, I have found amynthus, next to brackish waters in the coast of Connecticut, and parts of Massachusetts. I don't know what their tolerance is for salt water. That's a great undergraduate research project. Yeah, maybe Anis might know the answer, or probably Erin. I don't know about their saltwater tolerance. That's a great question.
Yeah, Justin, there was some good conversation yesterday. One of the participants had said that they sent Hillary and myself a video of jumping worms actually like moving through a bucket of water, and they seemed to be okay. Not that they were able to escape necessarily, but that they were not dying right away. Do you have experience with understanding how long it takes if they were to drown in a bucket of water? I usually tell folks to do some soapy water, and that helps to kill the worms. But if it's just water, are they fine to thrive?
Yeah, great question. So earthworms live in soils. Soils typically have CO2 concentrations like 4,000, 5,000 parts per million. So for them to suffocate in water is actually pretty difficult. They will eventually drown, but it's a pretty slow process.
Pretty
Yeah, so
slow
I think the
process.
soapy water vinegar will make it a much faster process. Yeah.
Yeah, okay. Well, that's good to know. I have another question in the chat here from Margaret, who says, I have used the soapy water method in a cut open milk jug for killing worms, but if you leave them too long, they start to smell very bad. I started putting them into the empty milk jug and leaving them in the bright sun to dry them out till I die. Should I not dump dried worms onto the ground? Would trash be better?
Yeah, so there's a great question. If there's questions like if you have the earthworms dying and they have laid cocoons or they're not all the way dead that they could potentially spread. As you probably smell, Amynthus and metaphyre earthworms are like pure nitrogen. They have lots of amino acids, so that could be good for soils. If you have soils that already have them, adding them back as nutrients, I'm not going to say yes or no to that. To me, biogeochemically, it makes sense. But, yeah,
Yeah,
I'm not going to go either way.
Yeah, it's a tough question. I mean, from my perspective, I would try not to be throwing any kind of jumping worms or associated materials back onto the ground or into a forest setting, just out of extreme precaution to try to not spread any of the cocoons if they're adults, or any potentially spreading that dirt that might be still attached to their bodies into a new area. Because again, these cocoons are really hard to see and very easy to spread, and that's not what we want to do. Stephanie asks, do these worms ever feed above ground?
Excellent question. So Stephanie asked, do these metaphyre and amynthus earthworms live above ground? And the answer is yes. I have found them living on logs, like the top side of logs when it's wet. They will kind of eat the microbial mat on logs. So yeah, I have found them crawl up inside of pots that I've had like potted plants on the ground that they will climb up there. So they're pretty good at climbing, especially if it's wet.
Yeah, that's terrifying. I saw them climb up a piece of cement block once. So knowing that they can do that is a little spooky.
Yeah.
When I was doing my PhD, this was just probably earthworms, I'm not sure, but we were doing stink bug traps that were, I think, 5 feet off the ground. It was like a cone on the ground that the stink bugs would land on and then climb up into the trap. And oftentimes those containers would be completely chock full, like to the brim with earthworms.
Yes. I see some question from Leslie. Will cocoons be killed if immersed in water? And I'm pretty sure the answer is no. I'm not sure about the submergence time. I wish Joseph was around, but maybe Erin in her talk will talk about it. There's going to be they're going to have to sit a while. You'll probably have mosquitoes before you can consider them dead by just sitting in water. And Amy had a great question about soil structure. Great question. So the Amynthus, the jumping worms, they make a very unique type of soil structure. It's like a very small, subangular, blocky, granular structure. So if you have any larger structure, it gets lost. Can you bring that back? And I'm going to say, as long as you have jumping worms, the answer is no. The structure is just going to be very different. Now, I'm not sure what the impact is on your vegetable garden. I have a vegetable garden that has a bunch of jumping worms, and I had one in Massachusetts that also had a lot of jumping worms. They seem to do all right. It seemed like they dried out a little bit faster than other areas. And then you can't keep mulch on them because they eat the mulch pretty fast. So yeah, your mulching is kind of the best that you can do, but unfortunately you feed them, so it is a vicious cycle. But by adding the mulch and organics for them to keep the structure, it's going to help fight it back. Yeah, I wish I had a silver bullet, and that's why we're here, but unfortunately I'm here to say, like, we need more research and time to figure out the answers to that.
It looks like we do have another question from a number, so I'm going to see if I can unmute that. So 207-481-1536. You can unmute yourself if you'd like to ask Justin a question.
Can you hear me?
Yes, we can hear you.
So my question would be in regards to like the water, how we learned yesterday that at 104 degrees, it's supposed to kill both the worms and the cocoons. Essentially, could we just maybe boil the water that the worms are in and take care of that problem? Does that sound right?
From a technical point, sounds great. From an actual practical, boiling water with the worms in there and cocoons and the horrible smell and the time to do that, it might not be the most practical, but it would work in theory.
Maybe people could toss it into their campfire.
Yeah, there you go. Let's see. Lee's asking about mycorrhizal fungi. I don't know if maybe I missed an earlier question or what that was in reference to. Oh, I see. Oh goodness, let me try to see it. Okay. You mentioned briefly impacts on fungi, and I'm wondering if there are studies on impacts on mycorrhizal networks. And also fungal related, do we know of a virus/fungi/bacterial bacteria that will impact or kill the worms?
Yes, I think there's a talk later today on control, and maybe the forest talk will be able to talk a little bit about the potential impacts on fungal networks. To the best of my knowledge, there's nothing on Metafire and Amynthus on fungal networks. Maybe Erin can correct me. I've seen some papers with some of the European earthworms, and I can't remember if it was like no significance, a mixed result, or a net loss. Unfortunately, I don't have that on the top of my head. If I find any papers like that, I'll send that to Brittany and Hillary to send out, but I don't know if there's any correct answers on that.
Yeah. Thanks, Justin.
And
So
one last point.
just, oh, go
Sorry,
ahead.
about the bacteria and the fungal. One of the things that we've been mulling over with the Healthy Soil collaboration is that if there's going to be any bacteria or fungi that impact the jumping worms, it needs to be jumping worm specific, and we're pretty far away from having any kind of biological control at this point, to the best of my knowledge. Again, I'm not a biologist.
You're darn close to one, Justin. Thank you so much for your presentation. Scale. Erin Cameron is a global change ecologist and associate professor in environmental science at St. Mary's University in Halifax, Nova Scotia. She has been working on spread and impacts of European earthworm invasions for the past 20 years, but only started to work on jumping worms several years ago when they were first found in Atlantic Canada. Erin, thanks for your flexibility today. Apologies again to everyone for the technical difficulties, but the floor is yours.
Great, thanks for having me. Yeah, so I haven't worked on jumping worms nearly as long as many of the other speakers. So my talk's definitely a small scale and short time frame. But if people have general European earthworm questions afterwards, I can also answer some of those. Okay, so I'm going to talk about jumping worms in Canada. Jumping worms occur in at least 38 American states at this point. They were first found in Ontario, Windsor, Ontario in 2014, and then New Brunswick in 2021, and Nova Scotia in 2022. So I'll be talking about our research in New Brunswick, in the yard where they were first found. So I'm gonna, I sort of thought that Justin would give a broader presentation, and I was correct. So I'm gonna really just focus on what we've been doing at this one site. So we had a few research questions. First, how do jumping worms affect European earthworms and soil properties at this small scale and likely over a short time period, because we think they arrived quite recently. Secondly, I'll talk about whether a wooden disc method that we came up with is as effective for detection as mustard sampling. And lastly, I'll talk about jumping worm spread at the local and landscape level in New Brunswick. So our study site is here, the red dot. It's near Fredericton in New Brunswick, which actually looking at the map is not very far away from some of the places in Maine that people have talked about. And the first place that they were recorded was in someone's backyard at this log pile. So he reported them in the fall of 2021, and then in 2022, we headed out there to try to do some research on them in that yard. So it's basically a lawn that we're looking at, which is a bit of a different habitat than other people. And in this yard, we set up this sampling grid. So the X in the middle, the red X, is that log pile. And then we have these sampling points, the black dots, that are 10 meters apart. So we set it up that way to kind of look at spread away from the log pile, assuming that that is where they were first arriving in the yard, which we don't know. We just know that they were very abundant there. These orange areas on the map are houses. The brown area is a place where the landowner had brought in some soil from elsewhere, which we kind of wondered might be another introduction source, but they weren't that abundant out there, so probably not. So at each one of those black dots, the sampling locations, we sampled in a 50 by 50 centimeter quadrat using mustard water. And this was an honors thesis from Sam Bennett, who's here. And in each of those quadrats, we also collected some soil samples. And then at each of those locations, we had the mustard quadrat, but also this wooden disc. So we'd observed, especially with that log pile, that jumping worms were really abundant under wood. And we'd found the same thing before with European earthworms. So we thought putting out discs might be another method of sampling for places where maybe like protected areas where you don't necessarily want to dig a hole or apply mustard, although the mustard is non-toxic generally. And we also thought this could be good for like citizen scientists or participatory science, where you could just put out a disc and then kind of forget about it, and you didn't need to get mustard or anything like that. As well, we were thinking that it could be good for cases where there's a slight bit of background noise, but minor, so not coming from me is the point. So yeah, we thought that the discs could be useful in cases where you want to look over a season at jumping worm activity, because you can flip it over multiple times without really disturbing the worms. And lastly, we thought that these discs might be useful at times when the soil is quite dry and the jumping worms might be less likely to be out and active in the middle of this lawn. Okay, so I'll talk about this first research question now, how jumping worms affect European earthworms and soil properties. There's been some research in other areas showing that there were negative effects on European earthworms. So these were, the figure there is showing where jumping worms occurred in the yard. So the black dots are locations where jumping worms were present. Red are where there were no jumping worms in 2022. So actually there's a lot more spread away from the log pile than we expected, but the pattern is consistent with the introduction starting there. So we found European earthworms in 26 quadrats, jumping worms in 18 of our quadrats, and we didn't find any significant effect of jumping worm abundance or biomass on European earthworm abundance or biomass. So no real effects here, but we think this is the invasion probably started only recently, so perhaps in the longer term you would see something. Then again, it is a lawn, which is different than where most other research has been conducted. Next, we wanted to look at soil properties. We looked at carbon and nitrogen specifically. Justin talked a little bit about that, or a fair amount, in his presentation. So we thought that there might be increases in carbon and nitrogen and like carbon dioxide loss with the arrival of jumping worms. In terms of carbon, you'd expect that jumping worm castings and burrowing activities can increase carbon mineralization rates, which might lead to greater carbon loss. For nitrogen, there's been some previous research that showed increases in total nitrogen content with jumping worms or no significant effects. What you can see with these percentages here is that we found basically the same mean carbon concentration in quadrats with and without jumping worms, and same for nitrogen. So no real effect of presence of jumping worms on either carbon and nitrogen. We did find a significant effect of jumping worm abundance on soil nitrogen, no effect of biomass though. So I'll just show a bit more detail on that one significant effect on the next slide. Here you can see soil nitrogen percent on the y-axis and jumping worm abundance on the x-axis. So consistent with some previous research, we see an increase in nitrogen with an increase in jumping worm abundance. But there's this one point N1, which was right near the log pile, which had a lot more jumping worms present and a lower amount of nitrogen. So it's hard to say with one data point there, but perhaps with higher jumping worm abundance, you might start to see a decrease in nitrogen content. We do have data from last year that we'll be looking at shortly. I forgot to analyze it before this talk, so maybe that will give more indication of whether this this point with higher jumping worms is not just a one-off, one weird plot. So overall, we saw limited effects of jumping worms on European earthworm. Well, no effect on European earthworms and limited effects on soil properties in this yard. Again, it's a short-term and kind of a different environment than other studies. So it will be interesting to see in the future what happens in terms of impacts there. All right, so our second question was comparing that wooden disc method with mustard sampling to see if that could be a useful method for detection of jumping worms. And so these were our results. The red is when jumping worms were detected with the disc, and the yellow is when jumping worms were detected with the mustard solution. So if you see red and yellow together, they were found with both methods. So basically you can see that there are some plots where you only detect with mustard and some where you only detect with the wooden disc. But overall, 14 quadrats had jumping worms with mustard and 13 discs had jumping worms. So there was no significant difference in detection, but the pattern was different. This kind of suggests that using both methods could be beneficial in some cases if you really want to get an idea of the spatial distribution. I think we potentially hope to do more research on this, looking a bit more at the type of log used and the weight and how detection changes with temperature and moisture before it, I think more research like that would have to be done before it would really be considered reliable as a way to sample jumping worms, but potentially an interesting approach in some cases when you might not want to use mustard sampling. All right, so move on to the last question that we had, which is how jumping worms were spreading at the local and landscape level in New Brunswick. So most invasive species spread partly by local spread, just moving on their own power. With earthworms, we generally don't think they can spread that much on their own, so there's no such thing as an earthworm stampede. The rates in the literature for European earthworms are about 5 to 10 meters per year for a population expanding. We did some work more recently in northern Alberta where we found we found an average of 17 meters per year, so it may be higher. There isn't much or any work on jumping worms. There's no work last time I looked, but haven't looked this year. So yeah, we just have these general numbers that it's maybe 5 to 10 meters per year or a bit more than that, but probably not super fast. So what really can drive spread of jumping worms is then long distance introduction events, anglers moving around bait, or potentially vehicles or the nursery trade. We've done more work on that with we've done a bunch of work on bait abandonment and transport by vehicles with European earthworms, but and starting to work on the water question as well with European earthworms, but we haven't worked on jumping worms other than this project. So here we tried to look at both the local spread and the landscape level spread. Yesterday, Josef Gorres talked a bit about why you'd want to know the distribution of jumping worms and invasive species. It's difficult to control if you don't know how they're moving around, and if you can't project future spread, it's more difficult to make a case for control as well. All right, so looking at local distributions, these are the data I showed you already for 2022, just in a slightly different color now. So the pink are the locations where we found jumping worms in 2022. Now watch this northward transect in particular, because that's what changes in 2023. Oh wait, I got that wrong. It's the southward one for 2023. Sorry. So we saw 1 point further, 10 meters further invaded when we went back in 2023. And we also sampled another point on the westward side just to see if there was the edge of the invasion there. That we can't really factor that into a spread rate calculation since we hadn't sampled there the first year though. Okay, so now in 2024, we see spread along the northward transect another 10 meters. Here, we couldn't sample the points closest to the log pile anymore because the landowner had built a shed there. So that's why there's some missing points there. And you'll also notice that there's more patchiness in the overall some points that had been had jumping worms in 2022 and 23 are now black dots without jumping worms. That's just kind of showing how variable the distribution can be from year to year. But basically, between 2022 and 2024, we see 10 meters expansion northward and 10 meters expansion southward, which potentially gives a rate of 5 meters per year. It is a bit tricky in a situation like this where the landowner is working a lot on the yard. Yeah, I wouldn't say this is a definitive piece of information on spread rates, but it does give us some data on local spread rates. Now that I've made this presentation, I think I will have to go back out this year and see if they spread any farther in those directions, even though we can't sample right around the log pile anymore. All right, so now I'll talk a bit about landscape level spread. We haven't done a lot of work on this, but we sampled at 50 locations across New Brunswick. We tried to sample near boat launches where people could be leaving bait behind, at community compost piles and near nurseries, and also in some forest stands. So there were 50 locations, as I said, which are represented by the yellow dots on this map. And what we found was that jumping worms were only present at 5 of the locations that we surveyed, which are represented by these red dots here. And one of those dots is the original yard that we were sampling in. Interestingly, all of the jumping worms that we found were at compost or nursery sites. So this does suggest that those are probably key vectors of introduction in New Brunswick. We didn't find any jumping worms near boat launches or in forest sites. And we did have a collaborator who sampled very extensively in forests across New Brunswick the last few years. So we looked at that data as well, and there were no jumping worms at this point in his data, which was good to see. Definitely more sampling across New Brunswick would be a good idea if we wanted to more definitively say what the drivers of spread at this landscape level are. All right, so just to wrap up, we saw no effects of jumping worms on European earthworms, some effect on nitrogen concentration, no effect on carbon concentration. The wood disc method seemed to detect jumping worms at a similar rate to mustard sampling. And jumping worms seem to spread at a rate of about 5 meters per year at the local level. At the landscape level, there were only 5 detections, all associated with compost and nurseries. So our next steps here are to continue doing outreach in New Brunswick and Nova Scotia. We had made these infographics with the New Brunswick Invasive Species Council, and whenever they've been doing outreach, they've been presenting jumping worms as one species that people should look out for, which has led to some reports, but in New Brunswick, none of those were actually jumping worms. In Nova Scotia, a couple of them were jumping worms, because we've also been working with the Nova Scotia Invasive Species Council and hoping to continue working to understand spread in Canada. We've been doing just a little research on cold tolerance with Amynthus tokyoensis. I forgot to mention earlier in the talk that there are 3 species that have been reported in this yard, but in all of our samples, we only found Amynthus tokyoensis. So basically everything I was talking about is about Amynthus tokyoensis. In this little study, we found that when earthworms acclimated to the summer were exposed to 5 degrees Celsius for long durations, survival was high during the first 6 hours of exposure, but dropped to about 0 after 24 hours. And then all the worms were dead after 48 hours. So essentially, they're not too, they're pretty chill susceptible. Anyway, I will leave it there so there's a bit of time for questions. That did take a little longer than I expected. If you have, you can email me there if you have any questions. And
Talk here. When do you leave? I met Terry at the.
Thanks, Erin. Sorry about that. I was trying to mute as people came in and left since I changed the audio settings to try to get Tricia's talk to work. So my apologies. Great talk, Erin. Thank you so much for that. We do have some questions in the chat, and if there are people who would like to also ask their questions, just use the raise your hand feature, and then we can go ahead and go that way. There is a question in the chat from Jacob, who asks, I think water washout downhill will move worms' cocoons with erosion during storms. I do wonder if jumping worms migrate uphill during storms like other worms that go to a high point.
Yeah, that's a great point. I have not seen anything on that. So I don't know, maybe someone else on the call can comment. The yard that I was talking about was completely flat, so we didn't have that issue here. There is, I guess, research on European earthworms that the higher elevation places tend not to be invaded as compared to the lower elevation.
Yep, I think that's helpful, and I can't say that I can speak to that question either. I have not really evaluated any research on where jumping worm goes, whether it's uphill or downhill, but thank you for the question, Jacob.
Yeah, it's definitely a good question. So just trying to catch up in case there's any others in there.
Yeah, there was some interesting conversation about worm grunting, which is, I guess, how fishermen use to collect bait. You rub some wooden sticks together and it stimulates rainfall to make the worms come to the surface. I haven't tried it, but it seems like it works, or it sounds like it works. So that's interesting.
Yeah, I guess you use a big stake to try to get vibrations, but I haven't tried it either.
Yeah. Another question from Jennifer here. Do they get as big in the northern parts, presumably with a shorter growing season?
Yeah, that's a great question. We didn't check yet. We did measure every earthworm, so I think that's something that could be looked at with someone else's data set. I don't know, maybe, yeah, I guess Josef would have so some of the research in that talk yesterday, the cocoon work was done in the same yard in New Brunswick. He didn't comment, as far as I can recall, on a difference in size, but that would definitely be something that would be interesting to look at. Yeah, I see another question on wooden. How long did it take the wooden disc to attract the jumping worms? We don't know because we put them out in June, and then the landowner put them out, and then we came back in August to do the sampling. So if someone was going to use this method, that's another thing that would be worth checking before you'd really want to use it. Yeah, the owner did have the let's see, he cut the wood on site. I think he got some of it from a bit farther away, but it was nearby and didn't have any soil or jumping worms associated with it when it was brought in. Okay. That might be about it for questions.
I think so. Oh, another one from Sarah. Does the mustard mixture harm other insects like spiders or ants, etc.?
It doesn't seem to. Maybe someone else wants to comment on this, but we sometimes see the ants like trying to avoid it as well, but they don't seem to be harmed. Yeah, I don't know if anyone else wants to write in the chat on that or whatever.
I've done a number of mustard pours, and I don't believe that it is hurting any of the other insects that are present in the area. The way that mustard works is there's a chemical component in ground mustard that's really irritating to the worms, specifically because they're invertebrates. They have this very slimy open body. And so when the mustard comes into contact with them, they will wiggle to the surface to try to move out of the way. Whereas other arthropods like spiders and ants and things like that, they have a hard exterior that is really protective. So they're not as sensitive to mustard pores in that way. If you are concerned, though, what I like to do, especially when I'm doing mustard pours in a forest setting, is I'll bring 2 jugs with me. One will be the mustard solution and one will just be clean water. So I will do the mustard pour, wait for the worms to come to the surface, hand pick or ID them, but then I'll also just flush the area with that clean water, just add it as an abundance of caution to really dilute the mustard present there. Lots of questions in the chat here.
I think I have got the I got Jennifer's. I missed the Liberty Library one. Yeah, that. Sorry, I also froze for a second.
Oh, sorry about that. Yeah.
So how does the homeowner think they got there? Yeah, they thought maybe the soil that was brought into that one area, it came from a military base, but we sampled that fairly exhaustively and didn't find jumping worms there. So we think it's probably from nursery plants was the most likely, because he had brought in quite a few plants to his yard.
Okay, and then I think if there was one more question, I'm my chat keeps on moving. I
think it's good. I can.
You think you got it?
Keep answering.
Yeah.
written if needed. So
Yeah. Okay. Well, thank you again, Erin. Really.
But yeah, let's just give this a whirl and see. This is me. I am holding on to. I worked in the natural areas for a little while before my current position. I'll talk a little bit about that in a second. And I am holding on to a tree, a beech tree that one of our famous educators, May Watts, had planted because she believed that our oak woodlands were going to convert to a maple beech forest. Well, she was half right. We have a lot of maple, we don't have a lot of beech. And one of the questions that I had when I was working is why the oaks weren't regenerating. So if you could go to the next slide. Here's a little bit about me. I am with my STEM internship group. If you could hit the next button. I inspire people to care and monitor trees. I encourage tree research, and I want to inspire the next generation. That's why I took on a 6-week internship program with them. We studied some of the original questions, the research that we had on jumping worms. Jumping worms are my passion project. I love jumping worms, and I love them because they're such an unbelievable. I don't love them, right? I mean, they're dreadful and I don't want them. But as far as a research project, I found it to be very fascinating. I am the Forest Pest Outreach Coordinator for the state of Illinois, and my position's funded through the U.S. Department of Agriculture. So their Animal Plant Health Inspection Service, PPA, which is the Plant Protection Act grant. And the Plant Protection Act really focuses on early detection. And so for me, talking about jumping worms and early detection and kind of like historically what happened and how can we prevent them and how can we understand them, I think really works together. So the information that you're learning today, you'll be able to, it's like a ripple effect. You'll be able to take it out and you'll be able to share it with your community so that people are aware of jumping worms. Spoiler alert, jumping worms are here. There's nothing commercially rated to kill them. I think it's something that we need to be aware of as far as cultural practices. And I think there's been so much consistent good information given out over the last 2 days that I think we're really well trained up. Next slide. We talk a little bit about the origins and the distribution. Great. So jumping worms has been spreading for 100 years. I know that we were talking a little bit about, I think in Justin's presentation, like how long have they been here? When was the first sighting? There are many different iterations of what was the first sighting, what was the first documentation. We know that our invasive species primarily come on, what we used to call maritime routes, so all those trade routes, all those ships. They've been spreading for 100 years, invasive species have been. And it's really spread due to human activity. So people who want, when they came over to this country, they wanted their plants, they wanted all of their animals and different, entities to come with them into the United States. And United States has been the one to really bear the brunt of it. The exchange of plant and specifically soil materials between Asia, Europe, Africa, and the Americas were the last. With global trade expansion, it became a high-risk pathway, and it still remains a high-risk pathway. Yesterday, one of the presenters mentioned that with all of the materials coming into the United States, it's less than 2% that actually gets inspected. And if you think about what we know now, right, of the jumping worms cocoon and how small they are and how easily they could go undetected, no matter how much surveillance you have, it would be very problematic to try to survey every single or inspect every single package that came in. With the advent of e-commerce, I think that has exacerbated the spread or has increased the amount of invasive species being able to come onto our soils. So that is a concern. People are aware of it, and we're trying to address kind of what are those e-commerce ordinances and rules that should be put into place. Next. A little bit of fun history for you. This beautiful picture on the right-hand side is from Japan, and it's in a forest. So the native range for jumping worms is basically Japan and Korea and a couple other areas. Amanthus tokiensis was found in Tokyo. So that is a little trivia for you. In Japan, they're not invasive. And it was fascinating to me to kind of understand, why aren't they invasive in Japan? And they are invasive here. They're in our hardwood forest here. And they are very, very, very aggressive as far as their population expansion, range expansion, and then their preferential feeding. In the forests in Japan, they have pine, evergreen oak, zelkova, ginkgo, and then the Hinoki cypress tree. All of those trees, the nutrients, that soil base, you think about pine, the pine needles, how acidic that isn't a very high beneficial nutrient pulse or soil nutrient, as opposed to the ones in the United States. So if you go to the next, if you think about our hardwood forest, what do we have? Maple, we have beech, we have basswood, we have all of these, what I would consider to be, and we have oaks as well and hickory. I would consider these to be high carbohydrate trees. Jumping worms, the complex, so you know the three complex, you have Agrestis, Tokiensis, and then Metaphyre Hilgendorfi. What, from my experience, I've always seen all three of them together, and I still don't understand why. But in these heavy maple dominated ecosystems, jumping worms can thrive, and they can stay there for decades. At the Morton Arboretum, we had forestry plots, and those forestry plots, the areas are dominated by maple trees, and it's a closed canopy. There's a lot of organic material. There's a lot of downed logs. We call those nurse logs. Jumping worms associates with lignin. So if you think about a downed log and that log is decomposing, and there's lots of different layers. You've got a leaf layer, you've got the protective layer of the decomposing log, and you've got that microclimate, you've got that lot of that nutrient, that moisture that they really need. So our forest composition favors their feeding preference. In southern United States, it's warmer, it's more humid. Obviously, the jumping worms are a little bit more tropical. They don't like high heat. They don't like drought. They don't like high temperatures. With our forecast, we're forecasting for warmer temperatures, and we can talk a little bit about the assessment that we did through the Illinois Invasive Species Council. There's a climate vulnerability study group that is working on assessing provincial ecosystems, so Canada through the United States. And one of the big issues for invasives are jumping worms. And so jumping worms, this conversation that we're all having right now, is very timely, especially as you think about the different models for climate forecast and temperature. Next. Here is a little bit of, I just put up this map, and again, Gary said they do find them in mixed wood stands. Yeah, absolutely. It's not that they won't go after oak or survive in an oak, but I think less of having jumping worms, maybe say in an oak or woodland savanna, where you've got areas that have high, grasses and different type of forbs and more sunlight. They're less likely to thrive and survive in that environment versus kind of, that maple beech dominated. This is a historical introduction to the United States. Currently, there are 29 states. There's one province in Canada. This should have a little asterisk to it. It changes all the time. I got this information off of EDSMAP. EDSMAP and iNaturalist are really only as good as the people that are actually reporting them. So in 1866, the first record was collected. It was collected in San Francisco, and it was named Metaphyre californica, which I think is kind of funny because they always name the species after the location, like Tokiensis. Jumping worms were first collected in 1939, 1947, and 1948. So Agrestis was found in Maryland, Tokiensis was found in New York City, and Hilgendorfi was found in Albany. There is some discussion that the first technical or first early record was also in Appalachia, up on one of the high hills, but I had trouble getting that type of documentation for this presentation. Next, please. In the great state of Illinois, we have over 50 counties that have a positive or suspected jumping worm population. We use this as citizen science, and I love Angie's exercise about breathing. I practiced that while I was trying to, um, uh, get logged back in. I just practiced her 4-second breathing. We do gather information and reporting through the University of Illinois Extension or the U of I plant clinic. I think what happens that during the growing season, when people see worms or they're active and they're out planting, they see a lot of material around, they get very panicky and wonder what it is. Many times we get really early season reports, but they are not jumping worms because we know that jumping worms are a late season species. We have done a significant amount of education outreach, but really where we fall short, certainly in Illinois, is doing any current, in situ. So where are those field researchers? I think sometimes with species like this, research needs money, and unfortunately for the Arboretum and for other organizations and possibly an extension, we just right now don't have those research dollars associated with this type of long-term research. Next, please. So how do they get there? We got human, we can go back a little bit, human mitigated dispersal. So international trade, I mentioned that, the transport. As soon as you see kind of those trade networks start to open up, that's where you see the increase in the amount of invasive species that are imported. Conversion from land to urban and suburban, it also spreads it when you're looking at developments, and people are bringing mulch, materials, soil, plants, materials outside of a local area. Landscaping practices, right? We have irrigated, we're composting, we're mulching, and then we want to get material that may or may not be native to our site. That irrigation, composting, and mulching, all good, solid landscaping practices. Do not get me wrong. I am a little bit unconventional. When people have a small infestation, one of the things we talk about is just pulling that mulch back. Don't overwater your yard. Don't overwater your beds. Take that composting out for a little bit. You can use composting tea instead of actually having that compost or that mulch. Mulch is like bringing the forest floor, but if you've got a small infestation and you want to try to knock it back, the best thing you can do is to use Mother Nature and to use a little bit of the sun. One thing that we're concerned about is the transport or e-commerce of materials coming in. Some of our areas in and around Illinois have a high increase of non-native material that has come in, and it's certainly come in off of state lines and whatnot. I think people need to maybe be a little bit more vigilant on buying plant material in an area where potentially they know that there's an infestation, or from areas taking mulch from areas that are known for potentially having it. So we have a municipal lot, and in that municipal lot, we know we have jumping worms. And so unfortunately, I would not encourage people to be taking mulch from that municipal lot. That mulch needs to be heat treated, and it needs to be heat treated over a long period of time. And we've talked about that previously. Next. Forest ecosystem spread, right? Primary introduction and entry points, firewood. Firewood is the #1 way that pests and pathogens move around the country. If there is one message that I would like everybody to really take hold, is don't move firewood. October is the National Don't Move Firewood Month, but I think that if we can all just focus on buying local and burning local, we would do ourselves a real service. Trailheads and mulched and managed trails. I put mulched and managed trails. If you think about an area that has jumping worms and the jumping worms, the cocoons might be in mulch, and you take that mulch and you spread it out over 9.5 miles of trail. Well, you've just distributed, potentially those cocoons everywhere. And so again, it becomes a challenge in making sure that we're vigilant in using mulch that is actually managed. Campgrounds, there's an influx. Marinas, we know that invasive species can come in through marinas. I know that there's been a lot of conversations about the water and the whether or not jumping worms will survive in water. From my personal experience and my research, they won't survive in water for very long. We actually used pans of water underneath our steel grate. We had tree seedlings that had jumping worms in them. And we thought if they ever escaped, they would just go through the grate, but they would hit that pan of water, and that ultimately they would die. So it's interesting. And we got that recommendation from the University of Wisconsin. They said that they use water oftentimes as a mechanism for controlling any escapees. And obviously our rivers, our lakes, and our ponds, anywhere you fish. I think that the horticulture trade was instrumental in bringing about this and spreading jumping worms, but I also think that anglers are as responsible, and not unknowingly. I think if people are buying something that they think is really cool that might help them catch, their bluegill or their crappie or some other type of fish, and if it turns out that it's not that beneficial, the worst thing they can do is just actually leave them. And intuitively people think, oh, worms, will be fine here, so they think they're doing a good thing. But we now know that leaving jumping worms behind is not a good thing, especially because we know that they're parthenogenic as well. Next. I put this up here from Violet Lee for University of Minnesota. This is just a macro cascade effect, right? We have seen all of these things happen when you get exotic worms in. We've got the facilitation of invasive, we've got disease dynamics, we've got plant community, changes in plant community composition, we've got productivity, soil and water quality, and then changes in carbon, and then also, changes in the disturbance regime. Next. This is where we get into the conversation about whether or not what you see is really what's happening. So when I was doing research and trying to pull together information, and certainly visual images rather than just a lot of text on a page, you see this picture and it says the worm front moving through a hardwood stand. Well, is that really a worm front moving through a hardwood stand? I mean, that's a pretty significant population that is highly active. And I think sometimes I put this up, and I certainly had challenged myself that when you're looking at something, you need to really understand, is this what is going on? It's very hard to tease out because you don't necessarily see, the species, right? You don't see it on the ground. You don't see, any other signs of any type of other invasives or deer browse or whatever. And over on the right, and this was a before and after, but is that really a before and after of the same site? I question that, and I'm bringing this up because I was changing my presentation from yesterday to today to kind of talk a little bit about what are some other things that we should think about when we talk about, the forest ecosystem and potential spread and ways to not spread. So compared to the effects of European earthworms, jumping worms have been less studied and they're poorly understood. And I think that has just been a consistent message. The recent discovery is really a lack of the taxonomic expertise rather than a new phenomenon. If you go back over 10 or 15 years, some of these species have been renamed, right? They went through and there's a whole key that has been updated. I think it was like 4 or 5 years ago that really drill down into, if you see this, then it could be that rather than what it was before. So they're able, through use of technology, to get very specific and to actually get better at identifying species rather than having kind of large bucket. And I still think that Amanthus agrestis and tokiensis and Metaphyre hilgendorfi, is such a large complex. And there are other worms, but necessarily we don't see them or pay attention. And sometimes they get just misidentified. Next. Erin just talked about the rates of spread. Again, put a big asterisk on this one. There's not a lot of research, that scientific peer-reviewed research that talks about this. I know Wisconsin's done a great job. Their average was about 12 feet in their site that they saw. Natural spread from doing a lit review, anywhere from 10 to 30 feet, short distance. Cocoon dispersal, obviously, we talked about firewood, we talked about mulch, we talked about, if you're out hiking and you got it on your boots, or you've got it, a dog, or any other type of animal that can move it around, up to 75 feet, not so sure. I would agree that jumping worms, cocoons tend to move downhill rather than uphill. If you think of like moisture gradients, typically on the lower side, especially the north side, you've got more moisture, and it's just more, it's a better habitat for them. And then obviously human assisted, 100 to 500 feet. I'm not sure, but that was really the only sound table that I was able to get to. So I put that out there with a question mark. That's something that we should be researching. And that would be the next research question that I would have, is how significant, how far and how fast do they actually travel? And do they need to have the right sort of setup? Meaning, if it's a dry, hot area, are they likely to continue to move, or are they going to stay where they're at and kind of devour the material that they've got, locally, rather than kind of moving around and sourcing other materials out? Next. Streams and rivers, I just put this in there because I know there was a question about it yesterday. They do facilitate the transport of cocoons, but honestly, I don't really know how far. We need to understand and predict spread into undisturbed ecosystems. I think that one of the things that we were looking at, if you're trying to figure out spread, is identifying an uninfested area and an infested area right next to one another, and then getting out and monitoring that over time, and really clearly keeping that site intact so you can do a multi-year study. If it is out there, I have not been able to find it, but I do think that the need for in situ, meaning on site, and especially in the habitat that's favorable, so that we can get an understanding of how quickly they spread based on moisture, based on material that's available, and also looking at fragmentation. How does fragmentation drive populations? Does it knock it back or does it increase it? Not so sure right now. And then from there, what are your management plans? I think the site that we're thinking about, it's a heavily maple dominated site. The understory is not that great. The soil is just your basic, soil that you would find with an average pH. There's nothing highly unusual about it, invaded or uninvaded. It depends on the tree species that are there. Opening up that canopy a little bit, thinning it out, getting rid of some of those maples, getting a little bit of light in, getting a little bit of that tiered, understory in there, getting some shrubs in there, diversifying that site might significantly impact the species. Maybe they wouldn't be that happy there. Maybe they would move on. Maybe they would not be very successful in their reproductive capabilities. Next. Drivers of spread, I touched on this a little bit. Moisture obviously drives horizontal and vertical migration. Horizontal migration for jumping worms, vertical migration for our traditional earthworms, our Lumbricus rubella species. Wet leaf litter is an attractant. Think about that downed log. Think about how you layer. In our site, we've got oak leaves. And so those oak leaves, along with that maple log, that oak leaf doesn't decompose within a growing season. It can actually hang out for a year or two. And you think about that being a roof, right? The roof over that house and that little microclimate. So those cocoons have everything that they need. They have protection, and then they also have the moisture that's needed. Mineral soil. There was a question earlier too about soil, sort of that organic layer, and they say there's some discussion that when you are in a droughty time, that the jumping worms can move on and feed on the mineral soil, so that layer just below. So that I thought that was interesting. Drought. It can cause aggregations around tree trunks. So if you've got a really hot, dry area, they might aggregate into tree trunks. Cleaning out down logs, if that's a management objective for your site, might also assist in keeping populations down a little bit. They like moist riparian zones. We have creeks, and we know that jumping worms are found right around those creek beds. And then their dietary flexibility. Jumping worms has a tremendously flexible diet, meaning they feed on lignin. So what is lignin? Any kind of downed and decomposing material. So their dietary flexibility, it contributes to their invasion success. Next, please. Fragmented sites are very vulnerable. There's low to no disturbance. Over on the upper right-hand side, you see the purple and the yellow dots. So purple is infested, yellow is not. That was our team that went out to look at the spread. We did a transect, and then we took a 30-foot tape and went off in the direct northern the directly north, I should say, and then looked at not only the vegetation, but the presence and absence of worms. And we did that really more as a vegetation analysis, and we were curious to see whether or not we had, any presence of jumping worms. It wasn't really the right time to be having the adults out, so it was very tough to find them. About 10 years ago, the Arboretum had what they call like an IMLS survey done. So there's over 300 stakes, I think, in about a 500-acre plot, and over half of them had presence of worms. But it wasn't presence of jumping worms. It was based on the soil structure. And we know that the soil structure is the unique identifier for a jumping worms complex. There was adequate habitat, there was protection from predators. So sometimes fragmented sites, they're vulnerable for invasion. I thought that was interesting. Next, please. Changes in species composition. We talk a lot about this. This is where the long-term studies need to be done. Beneficial, like jumping worms are beneficial to invasive grasses, right? That gets in, they work up that soil, the seed comes in, whether it comes in from an animal, it drops down from the tree, it comes from a bird that just ate buckthorn, you don't know, right? So that soil is really open. It's really easy to have, the seed take hold and get that first year to 2 or 3 years of growth that they need without doing a lot of work. Ferns potentially could benefit from the introduction of jumping worms. There's some literature that highlights that. We know that there is an increase there is reduced plant diversity. There has been 1 or 2 studies that have been documented that they've seen an increase in sedges. And I think that if you have a healthy sedge population, that having this friable soil would contribute to the spread of sedges. Obviously, garlic mustard and then Japanese barberry was highlighted as well. Next. Soil characteristics. having co-occurring factors. So having, invasive plants, deer herbivory, forest age, land use. I mean, when you have jumping worms and you have different changes within your site, is it only the jumping worms, or is it because of deer, or is it because of the forest age or the land use, right? What is actually going on? What is co-occurring at that site? I think sometimes we're looking for that answer. And if we see something that, like jumping worms, and we don't see a lot of plant diversity, we instinctively say it's due to the jumping worms, or the presence of the jumping worms. I think earthworms have done a lot to work over the soil. And oftentimes you see this, the first wave of non-native invasive earthworms that come through. And jumping worms are like the second invasion, right? There's a great lit review paper, and I think it's called the second wave invasion. And it's a really, really good paper. But I think that sometimes we need to understand and kind of unpack what's going on a factor, and at a site. Next. I want to get down and finish this. Research motivation. So I was questioning whether or not what happened in the woods. Go ahead, keep going, keep going. Let's get through this. So you can go ahead and click everything. So in all of the literature that was available 5 years ago, it said that Amanthus species had unique soil signatures that altered the wildlife habitat and inhibited understory growth. It altered soil nutrient and eliminated leaf litter, and it reduced soil moisture, and it reduced tree seedling growth. Next slide. In the research that I conducted for my master's, we did see a significant soil signature. This is only about 2.5 months, if you will. And that was just, anywhere from 4 to 6 jumping worms that were in a pot. And we duct taped it and we kept them in there just to see what they would do with with the soil structure. And it was unbelievable. And I think that that is what is the greatest concern from my perspective on a long-term basis. What is this soil going to do? What is the trajectory of this soil, and how will it impact our forest, the health of our forest community? Next, please. So the results of our research, it did not alter wildlife. Next, just keep hitting it. Didn't inhibit growth, didn't reduce the leaf matter, right? It didn't eliminate all of the leaf litter, but it did increase decomposition rates, and it did have indirect effects below ground. But that was because, and then the other altered soil nutrient poise and the reduced tree seedling growth, it was site and soil specific and tree species specific. And I'm not going to get into the research that I did, but basically what it did is highlight the effectiveness of jumping worms on maple seedlings and also invasive species like buckthorn. Next, please. We need more in situ research. I've already talked about that. Next. So looking at the changes, looking at alternative states, we have, we need to figure out what's going on. What models can we use to look at a 2-stage invasion? I think this is really important. What is the second wave doing? And what are the impacts of co-occurrence? Next, please. We need models. Rates of colonization, changes in carbon storage, obviously life cycle change and temperature variations, and then moisture availability, range distribution, how far south and how far north can they continue to exist. If you're in the southern part of the country, you can have longer generations, you have longer growing days, you have more humidity typically, and you have warmer temperatures. So is that a contributing factor to expanding range? I don't know. I would say it could contribute, but research is necessary. Next. What can we do? We talked about this, and this this was already answered so many times in everybody's presentation, which is fabulous. Spray down your vehicles. I love Play Clean Go. June is National Play Clean Go awareness. So definitely trailheads, install boot brushes, and remember, do not move firewood. Next. Cultural practices, raking or blowing leaves where it's possible, making sure that when you are doing plant sharing, you are doing it properly. And there was some great information that was shared on how to do that. Getting those dipping buckets, I think, is really helpful. And then replanting it in a soilless mix for a little while is also beneficial. I'm not going to talk a lot about prescribed fire. There is some research. We need to do more research. Bradley Herrick and Marie Johnston worked on that. There's a paper. Using fire as a tool for management for the species, I don't think is beneficial. And we could talk a little bit more about it. The time in which you need to burn, and certainly burn in these areas, is not very doable for many sites.
And Tricia, I just want to point out it's 3:30, so I just want to wrap things up.
I'm zipping through.
Awesome.
You know what? We can finish it up. Quick. Good. We have an Illinois jumping worm task force. Yay! Next. Yep, we have done an assessment on jumping worms. So if you are interested in looking at it, you can check out the Illinois Invasive Species Council page on the Illinois Department of Natural Resources. It is supposed to be public. If it's not out there, I'll definitely double check after this presentation. Next. I want to thank my research team. They're fabulous. Next. That's it. Thank you.
So our next presenter is Dr. Vera Krischik, and she is a University of Minnesota professor and extension specialist in integrated pest management. She has 34 years of experience in pest control research in settings ranging from lawns to large restorations, with a focus on reducing non-target impacts on pollinators and beneficial biocontrol insects. Her lab has been requested to provide guidance on ecologically sensitive pesticide practices for the Minneapolis Park and Recreation Board and several national committees. Without further ado, Vera, if you'd like to go ahead and unmute yourself and share your screen, we can go ahead and get started with your presentation. Thank you.
Oh yeah, that's fine.
All right, so sorry about that. We found out the other day that there is a problem with UMN computers accepting Teams. So we tried to do a diagnostic session, and at least we got it so you can see something. So that's good. So my role with this jumping worms group is, for years I've worked on trying to figure out if neonicotinoid insecticides affected native bees. And so I'm quite used to doing bioassays that are acute and chronic. And so I just want to give you a caveat before we start. So I'm going to talk to you about all the work I've been trying to do, trying to get these chemical companies to work with the EPA and the IR4 to register a product for jumping worms. And it's not that easy because they don't want to do it unless they can guarantee themselves that they're going to make money. So the first issue is that they want us to register it for 3 species of earthworms, not just jumping worms, so that golf course managers could use it on greens to control any earthworms, because there are no products registered by the EPA for earthworms because the EPA considers them beneficial. You know, in Minnesota here, we don't consider any invasive exotics. Even European earthworms is beneficial because of wheat rhylex work. So anyway, from the EPA perspective, earthworms are good. There are no pesticides to manage earthworms. So starting down that tunnel, you have to ask the EPA what chemicals potentially might they register. You have to talk about what amendment to FIFRA that you can use to get it registered. And then you have to talk about how you collect efficacy data. And I've noticed a lot of people have asked about phytotoxicity and non-target effects. And so we're aware that those 3 sets of data are needed to give to the EPA to register any potential chemical that might manage jumping worms. So that's just an overview of what I'm going to talk to you about. So I'm focused, we're working on bioassays right now, trying to sort out what bioassay options we have that might work. And so we've been relying a little bit on the Japanese beetle harmonization program, which is a quarantine program for managing Japanese beetle that allows you to add insecticides to pots. And so it's similar to jumping worms that are getting into the nursery industry in pots, or jumping worms that are in mulch piles, either mulch piles in the greenhouse that might have Japanese beetle, or mulch piles that might have jumping worms in the field. So we used bifenthrin as a conventional toxic insecticide that is already registered for Japanese beetle. So that's basically our positive control. If we can get bifenthrin to work, and it should penetrate the depth of a pot or the depth of a mulch pile, because that research has already been done by the chemical companies. So to compare to that, we're trying to some organic or OMRI, Organic Materials Research Institute registered products. And we're mostly working with saponin and soap-like products. So that's kind of an overview of what I'm going to talk to you about. I don't have that much time, so I'll probably skip some slides or talk from New York, do my New York talk, which is very fast. But basically, our lab is working on this right now. They might be listening. They're doing experiments at this moment on jumping worms. We got some state money on trying to sort out just what I've said to you, working as a liaison between the state, Minnesota Department of Agriculture that registers pesticides, EPA, and the IR4 program, which is the program that works with manufacturers to get the data, the three kinds of data we just talked about, for managing jumping worms. Okay, so for our perspective, jumping worms are an invasive exotic that need to be managed because they're affecting forest health. And one of the things that we have noticed is that jumping worms emit a yellow fluid from the head and from the segments. My visiting scientists from India and some people in the lab first noticed this because we and others who have tried to do bioassays with jumping worms, you get very high mortality in the controls. You go out in the field, you collect 100 individuals, and the next day you only have 30. So I'm just telling you this up front because if any of you researchers or any of naturalists have noticed that, this seems to be some anti-predator chemical, that because you're keeping them in high populations together, it's affecting inter-specifically within the same species, intra-RA, but it probably has evolved for interspecific use. But that's one of the problems we're having with managing jumping worms in these bioassays, is the mortality is high. So we're using red wigglers and earthworms based on the fact that the label that would be produced would be for earthworms, not just specifically jumping worms. So some of you have brought up calendars and maps of where jumping worm is, the ontogeny through the season, when they appear, when they make their cocoons, when the adults die. And we can confirm that even though we kept jumping worms in the lab, all the adults die in November, and next March the cocoons start hatching at a lab that's 70 degrees. So that was one of the things we were trying to figure out. If we kept them in warm labs, could we keep them for a longer duration? And it looks like this diapause that they have, or this annual cycle, is actually, genetic. It's not flexible. So here's just, I put up some nice map of where jumping worms have been discovered in Maine, because you guys are from Maine. I'm sorry to say we don't have good maps like this. The other two sites that have been discussed, EDNS and iNaturalist, they don't have that many entries. So this is something we should work on, how ubiquitous is this phenomena going on. This is something I got from EDD maps, and it's really not that helpful. So we do need to spend a little more time looking at distribution. So in this grant, as I said, there's three components. It's already 2:42, so I only have a few more minutes. We want to determine pesticides, the chemicals, and cultural management. So we add chemicals because of mustard. We want to look at regular mustard drenches, hot mustard seed drench. If we can use something at a lower concentration to get them to the surface. We're looking at non-target effects. So we're going to do sosids, sow bugs, bumblebee colonies, all kinds of things in the soil. A number of you have brought that up. And we're going to look at phytotoxicity. We're going to use hostas and composite plant to see if the effects on roots or above ground foliage of these treatments down the road when we come up with what we think are going to be successful treatments. And we're working we've been talking to the EPA for 3 years. The grant just started last year. by trying to sort out the registration process and how for us to get information. So this is the part probably that's novel for this workshop. So we're looking at two natural chemicals of saponins. Saponins is a chemical class that means soap-like, and it will dissolve the mucus. So it shouldn't have non-target effects like people said earlier on crabid, staphyliniid beetles that have a hard exoskeleton without mucus on the outside. So it should be mechanistically specific to earthworms, which is what the EPA wants you to register. So they have suggested two recent products have been registered, and one of them is tea seed meal. And it's from Camellia sinensis, so from a very close relative of flowering camellia. In China, they use the seeds for oil, they press them. And then the ovules and the bracts and everything are left over, they compost it, and they make a soil additive that has very low nitrogen, phosphorus. But they sell it, and it's available for us. And this is the product that golf courses around the world have used for years, tea seed meal. It's got different names as companies go in and out of business. And that is a solid. And so we have data right now that shows 10 grams in a 3-gallon pot, or 10 grams in a bioacid container that's mimicking a 3-gallon pot should kill the jumping worm. So if you just use the raw product. But this tea seed meal should get registered by the EPA if somebody is willing to do it, because it's used as animal feed, it's used in cosmetics, it's used as into soaps that you call natural soaps. It's actually in the COVID vaccine. It's a foamer for pesticides and those inert ingredients. It's an adjuvant. So it has a lot of positive human health effects. So does vitamins. So there's a good chance that that product eventually could get registered. So again, they press the seeds. It would be just garbage, but now they're composting it a bit and selling it in these 25, 50-pound bags. The other, and so this is the one you've seen, Planet Turf is the granular. A company in Switzerland has recently come up with a product that is a liquid of tea seed meal. And we're trying to get a hold of that, and that would be better to use. It's not approved by the EPA yet. It doesn't have an EPA registration number, but it is a liquid form. And the problem with saponins is when you inject them in the machine, most pesticides, there's an active ingredient like bifentrin is a pyrethroid that has many different trade names, but that's in the Japanese beetle harmonization. When you try to do tea seed meal and you inject it in a machine to look at the peak that would identify the activity, it all comes to a problem because it fractionates. And saponins, because they have a glycoside on them, that is known. So that's why they allow them to register them as only saponin, which is kind of different. So here they say the mode of action, it controls nematodes, root knot, it controls egg hatching. And so the reason why EPA is suggesting this is nematodes also have a mucus-like membrane. The second product we're looking at is from soapbark tree, Cahia saponaria. And that is already registered as Monterey nematode control. And so we're going to use this product. Again, it eats away at this mucus membrane. And it's just this Cahia saponaria, this tree called soapbark tree. And there's two different companies that are registering products from this Cahia. Again, this is a saponin that they're registering. And again, this is used throughout agriculture, pharmaceuticals, different uses. It's actually eaten as vitamin. So again, the chance of getting this registered is very high. It has OMRI, Material Research Institute, approval. The other product, Vegilab product, also had OMRI approval. So these are the compounds we're working with along with regular insecticidal soap to look at the phytotoxicity. So this is just a bioassay of what we're doing. We're using as again this bifentrin. We're doing the Qualipro brand because it has a caution label and some of the other ag uses are very toxic. We're using the tea seed meal and then the liquid form, Monterey nematode control. We're also using a fungicide that people have shown as effective and regular soap, and then we have mustard. We are going to use regular mustard and very hot mustard, see if there's any difference. And then here are our two plants that are going to eventually register phytotoxicity with Gaillardia and hostas. And we're actively working with this IR4 program, trying to figure out what data they need to then work with a registrant, a chemical company, that they think there's enough market for this, again, for earthworms in general, to get it registered. And within the state, if there is a product already registered, like that nematode, Monterey nematode control, then you can add this local needs section 3 label. So that might be the first product we get registered. Just because there's already label, you don't have to register a product. We're just getting an amendment to that label based on our 3 tests, the efficacy test, phytotoxicity, and non-target effects. And so again, one of the things in order to do bioassays is you have to kill things alive. And, you know, we're doing bioassays that are short 94-hour bioassays, which is the traditional. We're not looking at LC50s. We're looking at label rates and how much above and below label rates you'd need to use. Anything above label rate, again, has to get a new registration with the EPA. And you have to keep these things alive. And as I said, we're having these problems with jumping worms that you touch them, they release this yellow fluid and they kill each other. And that is not much in the literature. I found an older paper that talked about this. So any of you guys who have information or are interested, we're poking them with plastic paper, all different things to see what we can use that we can keep them alive. And this has been a problem with many people's bioassays. So what we first did when we had 7 treatments and we were trying to figure out what for these short-term bioassays can we feed them so our mortality in our controls is 0. So just to show you, we've come up with, this is our jumping worms, this is red wigglers, these are night crawlers, and usually this number 7 comes up, and what it has in it is it has purina, it has worm food. It has actually either purina chicken feed or Memes worm food, and they seem to have the same result. So if you add mushroom compost, manure compost, core, here's the oyster shell, and then if you add this worm food, you get really good growth. Their weights increase, they don't die in mortality. So we have a way. We're very interested in the chronic studies where we want to not only do a 70 to 96 hour study, but we want to keep them in there for 5 weeks and see if the cocoons hatch, if the hatchlings die. So chronic studies are really important for, especially if you can use this out in the environment. So that's as far as we've gotten. I've left some time here for questions, and hopefully by the time we have another meeting, another workshop, we'll have some more answers for you. Are there any questions for me?
Thanks, Vera. If there are questions, you can feel free to unmute yourself or raise your hand and ask them. As far as the chat, there's been a lot of good conversation again. And Tricia, thank you for answering some of the other questions. There's a question from Sarah who asks, I've noticed jumping worms concentrating around the roots of transplanted seedlings in my garden. I'm thinking of putting mulch or leaves in one area of each bed to attract them and then hand picking them out. Do you think that could work to draw them away from plants, or is that wishful thinking?
Well, you know, we've been trying to focus on that too. We've been using green sprouts, lettuce. Some people use basswood leaves, leaf rye, who's like Mr. Worm in Minnesota. He says that's their favorite food. Erin Buchholz at the Arboretum says that too. But we don't want to bring dirty leaves in the lab. So I think leaves would work, but they seem to have a preference for basswood leaves. Just so if you're going to get leaves, get some linden leaves. That might work. I would try that myself. And we do think that they're feeding on roots. We're getting this feeling because they like the bean sprouts, they like the lettuce additives, that these guys may be root feeders. Maybe on root hairs, which is going to be a little problem in how you're going to quantify this in a phytotoxicity study. But we're with you. There's something going on with, you know, seedling establishment, seedling mortality with jumping worms around.
Maud asks, diatomaceous earth worked into dirt around the seedlings? Is that how you would go about using that?
So I'm just based as an entomologist. I'm going to answer this. Diatomaceous earth kills things by being rough. And as soon as you get it wet and put it in soil, it doesn't work really well. It works well on leaves and things that are drier because the sun dries them out. And it works very well in like a food warehouse where it's very dry. You put it on the bags of food. So I think it has a real important place. I don't think it's going to work for jumping worms based on my information in my brain about how the mode of action, but maybe somebody's going to correct me and say, oh, it works beautifully.
We did have a speaker yesterday that that said that diatomaceous earth was not effective for jumping worms. So
Right.
yeah,
I was
there's that too.
Yeah. Logic works sometimes.
Yeah, that's it.